Good morning, everyone, and welcome to the third annual Advances in Plant Breeding Symposium. My name is Dave Bubeck. I'm from Corteva Agri-Science, and I'm also the president of the Seed Science Foundation. So the session today is being co-sponsored by the Seed Science Foundation and the National Association of Plant Breeders. This is actually the third installment of a symposia series that we started back in 2020. We had one, and I've had the pleasure of being at all three of them, and these were designed to provide information sharing on plant breeding and plant breeding related technologies to the policy agencies, and encourage networking and interaction among us. And so we had the first one back in February of 2020. It was actually one of the last trips that I took before the beginning of COVID. And so then we had that in person in Washington, D.C. And then the second one we had was in 2021. It was entirely virtual in September of 2021. And we had a part one and a part two that were spaced about a week or so apart. And then this is the third one. We didn't do anything in 2022 in this regard with this series. And so we're restarting it here today and decided to do it again virtually. So next slide, please. So I already said I'm president of Seed Science Foundation. I'm also a research director at Corteva Agri-Science. And I'll be the moderator for the morning session. And then Thomas Luberstadt, who is also with us, is current president of the National Association of Plant Breeders, and he's on faculty in agronomy at Iowa State University. Next slide. So just a bit on the mission of Seed Science Foundation. We are a relatively new organization formed under American Seed Trade Association. But we have a long history in a couple of previous organizations within ASTA that merged together. So the goal here with forming the Seed Science Foundation, the mission was to address seed and plant science challenges and encourage plant breeding education and seed research. And the needs of the global seed industry are changing constantly, and certainly climate change and many other things impact the changing needs of the seed industry. So we identified 6 subject matter areas that were of key interest to the Seed Science Foundation. One of those was seed applied technology, digital agriculture, seed health and plant pathology, seed production technology and equipment, seed quality and testing, and last but not least, and why we're actually here today, is plant breeding systems. And so talking about this is a key area for us, and this is one of the things that Seed Science Foundation has taken up as a sponsorship to how do we educate those around us in regards to plant breeding systems. Next slide. And then the second group that is sponsoring for today's session is the National Association of Plant Breeders. National Association of Plant Breeders has been around for a couple of decades and came about as an association that brings together plant breeders from all different species, anybody doing plant breeding and crop performance improvement. The goal was to strengthen plant breeding to promote food security, quality of life, and a sustainable future. It's North America focused, but we have many members also from outside the US and Canada, and really leveraging knowledge on science and technology efforts to improve plant performance. That is the overall mission of NAPB. We do a lot about advocacy and spreading knowledge about plant breeding and the technologies and sciences that help foster a strong plant breeding program. We have a very diverse membership, and we cut across both public and private sector scientists. And great relationship as we build graduate students who are, working to become eventually career professionals. So there's a great interaction with NAPB and the student population that becomes student members. Next slide. Okay, so that's the 2 organizations that are sponsoring the event today. our objective for this whole series of symposia is to provide opportunities for individuals in U.S. and we've today, for the first time, we've added some Canadian policy agency members to hear from scientists involved in plant breeding and related technologies that contribute to improved products. The first two sessions, we were mostly focused on plant breeding and some of the fundamental technologies that have historically gone along with plant breeding and improved crop performance. And the session today will be much more focused around specifically genome editing. So the vision of this symposia series was to create a forum where you as the audience, the agency personnel, could interact and establish dialogue with technical plant breeding experts. And so we encourage the session today. We want a lot of interaction. We encourage questions and answers. Put your questions into the chat. We'll have moments after the each presentation and a panel discussion at the end where you can freely ask questions. We are going to record. We've already started the recording. We're going to record each of the presentations, but during the Q&A dialogue, we'll stop the recording and hopefully have some free and open dialogue. And you're welcome and encouraged to ask any questions that you would like. Next slide. So we had a change on the agenda this morning. So we are actually only going to have two presentations. We had some unfortunate miscommunications, and so the agenda that you might have seen that Norfolk Produce was going to talk about purple tomato, they are not going to be here today, so that's unfortunate. Don't blame them. It was a series of miscommunications, but unfortunately they're not going to be able to join. So we're going to have probably a little bit of an abbreviated morning, which will certainly free you up to ask more questions of the first two speakers. And so we may take a little bit longer lunch break, and then we'll come back in the afternoon, and then we'll have 4 talks in the afternoon session, which Alan Van Deynze will be the moderator for that. So I think one more slide, maybe. Go ahead to the next. Yeah. Okay. So thanks for attending today. I hope you really enjoy this session. I look forward to some good dialogue along the way. And I'll also, just a placeholder, we will be sending out a survey after this. We really want your input as to what's beneficial for you guys in the agencies. How can we make these better? We're talking about potential in-person events in the future, and we just want to make these beneficial. They're really intended for your benefit and give you an opportunity to network and get to know many of the people in the plant breeding community across both public and private sector. So with that, I'm going to introduce Nicole. Nicole Juba is with Pairwise, and you all hopefully know about the work that Pairwise has been doing, and really looking forward to Nicole's talk. Nicole is an associate director on the regulatory side with Pairwise, and she's going to talk about building a healthier world through better fruits and vegetables. Nicole?
Yeah, thanks, Dave. Good morning, everyone. Thanks for the opportunity to join you all today. Really excited to share with you how Pairwise is going about using new breeding tools to deliver better fruits and vegetables to the U.S. consumer. I joined Pairwise about 2.5 years ago because this mission really resonated with me of healthier options for people with very busy lifestyles. For those of you who aren't familiar with Pairwise, we are an all US-based company in the food and ag tech sector. We have our headquarters in North Carolina, along with our main lab and growing spaces. We also have facilities out on the West Coast where we do production optimizations as well as field work. So we have coast-to-coast expertise and a talented group of about 160 people bringing these innovative products to market. Backed by a who's who of founders from industry and academia and reputable organizations like MIT and Broad Institute and Mass General. We have access to technology and talent in the CRISPR tool, the gene editing space, including licenses for use of these technologies in agriculture, as well as our own proprietary tools that we are developing to enable more efficient editing for targets for different crops. As we build our business, Pairwise leverages our own internal expertise, as well as partners with others throughout the value chain, which is very important to our business model. We like to think of this setup as a barbell, where Pairwise is heavily involved on the front end with the genetics, the tool developments, the registrations where applicable. And then in the middle, we partner with seed producers, plant propagators, produce growers, produce packers and shippers. And then we come in heavy on the back end, where under our own brand, Conscious Foods, that's Pairwise produce brand, we work with food service and retail partners. Food service is things like getting our greens into restaurants and cafeterias. Retail would be getting our products onto the grocery store shelves that you shop at every weekend. And then again, that brand being the face to the consumers. And we think this is a really unique model so that we can be close to both the science and the innovation at the start, as well as the consumers on the other end, and really gauge that feedback and be able to take it right back to the next products we're developing. The products that we're working on are using these new breeding techniques like CRISPR to unlock the genetic diversity that exists in specialty crops. We do focus at Pairwise on fruits and vegetables. We do some work in row crops because of partnerships we have with other companies or developers, but our branded focus is on fruits and vegetables, those specialty crops that have maybe missed out on some of the advances in agricultural development over the last several decades, things like transgenics, and haven't been able to advance maybe quite as far, have a great opportunity as we use things like CRISPR. And again, we're looking to take what's there in the diversity, get there faster, and get these great products to market. An example we like to share of how we're going about doing this is the story of the pitless plum. Pitless plum is a fruit that has been developed through breeding to not contain that pit in the center. It would be possible through centuries potentially of testing and crossbreeding to take that pitless phenotype in the plum and breed it into cherry and create a pitless cherry. With gene editing, we're able to target those mutations that could have been produced through breeding that exist in nature and create a pitless cherry product faster. This is an example. This is our target. This is where we want to be able to head with these new technologies. And one of the reasons we're doing this and we're focused in consumer crops like cherries, like salad greens, is we know consumers want to see more variety and more option in the produce section. And we know as we have more innovative products, the consumer responds to these. And innovation can be something like year-round production. We saw a huge increase in consumption of blueberries in the United States as we were able to have a year-round supply of this berry. People are interested in it. If they can have access to it year-round, they're more likely to buy it month after month, week after week in the store. We also know other products like baby carrots and seedless mandarin oranges. These are snackable products. You can put them in the car, you can put them in your bag, you can take them to soccer games. We live very active lives where making the healthy choice is sometimes difficult. So at Pairwise, we're looking to break down that barrier by making the healthy selection an easy and enticing choice for consumers to make. And that leads us to our very first product. We are the first in the market with a branded gene-edited product under the name Conscious Greens, and it is a gene-edited leafy green. At Pairwise, we saw an unmet need to have a nutrient-dense salad option that was very attractive to people and to consumers. And we could have gotten there in a couple of different ways. We could have taken the standard salad base, like romaine lettuce, and modified it to have a higher nutrient value. That's a complex system, but we could have gone about that route. Or we could take a leafy green that was already naturally nutrient dense, but maybe had an off-putting flavor like kale or mustard greens, that when you consume them raw, they have this pungency, this bitterness. It kind of tastes like wasabi. We took that route where we took an already nutrient-dense leafy green and were able to edit to improve the flavor. We were able to create a naturally occurring mutation in our preferred varieties and improve the flavor. For those of you who might not have as much experience with leafy greens or brassicas, brassica species in general have a wide diversity of leafy characteristics. They come in vibrant colors, like deep greens and purples. They come in various textures and shapes, and we think they make a really attractive option as a salad base relative to some of those more common lettuce options. At Pairwise, when we talk about working in brassica, we're specifically talking about Brassica juncea. And even within Brassica juncea, we are specifically working in the varieties that are traditionally used as leafy products, either baby leaves or mature leaves, but they're intended for leaf consumption. There are varieties of Brassica juncea that have been bred for canola use or spice mustard use. Those are not the varieties we're working in. So we're taking the great diversity and nutritional value of these varieties of Brassica juncea that have been bred for leafy use and gone about to improve the flavor. We've done this by reducing the pungent compound found in mustard greens. Mustard greens, like brassicas, contain glucosinolates. The main glucosinolate in Brassica juncea is a compound called sinigrin. Sinigrin accounts for almost 100% of the glucosinolates profile in Brassica juncea. When you eat these mustard greens, an enzyme called myrosinase is activated, and it is going to break down sinigrin into glucose and this pungent compound, allyl isothiocyanate, AITC. This is the compound that's creating that mustard flavor, that wasabi-like effect when you eat raw, uncooked mustard greens. There exists a natural mutation in brassicas that creates a loss of function in myrosinase. And that's what we have replicated in our pairwise mustard greens. By creating a mutation in the myrosinase sequences, we no longer have this enzyme, so that when you chew the mustard greens, sinigrin isn't broken down into this pungent compound, and instead you have this really nice flavor profile in a product that was already naturally nutrient dense. In order to go about creating this product, we had to target very specifically the myrosinase sequences in our Brassica juncea varieties. We did this by designing guide RNAs coupled with a high fidelity Cas12a. And in doing this, we needed to target 17 myrosinase sequences in our mustard's genome. We were then able to self and fix our edits in our final product and segregate out the foreign DNA. We did use a transgenic intermediate. We then followed line selection of our edited material, just like any conventionally bred Brassica juncea for produce use would have done. We checked to make sure there were no off-types. We also characterized the edits in our commercial lines and demonstrated absence of that foreign DNA. We've published all the information about how we designed our constructs and our editing tools, how we selected our materials, and how we screened it to make sure we were indeed getting that low pungency phenotype at the end. And the references here, we published this last year in the journal Plants. Now that we have created this product, it was important to Pairwise that we get some consumer feedback. Our mission is to produce consumer-centric products, things that are going to resonate with the consumer. So in order to get the consumer's take on our gene-edited, low-pungency, nutrient-dense mustard greens, we went out last summer to various street festivals in the US, one in Seattle, one in Palo Alto, California, one in Austin, Texas, and we offered people the opportunity to try our Conscious Greens salads, our branded gene-edited salad product. In having these consumer engagement opportunities, we were very transparent about what we were offering people the opportunity to sample. We did this through signs and placards, QR codes. Our ambassadors verbally shared with each guest, this is a product of CRISPR, this is how the salad was produced. And at the end, we asked consumers to give us the feedback on our gene-edited salad. Of the 6,000 salad samples we offered at these 3 festivals, 3,000 consumers, so roughly half of the people, responded to our online survey, which was a very good response rate. And 2 key things coming out of these surveys. One, the intent to purchase our salads was high. Weren't able to show some of the videos today, but we would interview people at these events. We would interview parents, children, people of all generations, and it was really great to see, especially children, excited to eat these salads. The other noteworthy survey result is that of the 3,000 people that responded to our survey, only 1% of people mentioned anything about the technology, and not all of that 1% was a negative comment about the technology. So we feel like now we have developed a product that meets a need, a product that has good consumer response, and we've used some real innovative technology to get us there. Just to show you a couple slides on what consumers were saying when we asked for this feedback, when we asked, what do you like about these greens? They say fresh, good, flavorful, tasty. On the other side, when we asked what you don't like, they said nothing. Some people said, well, it's still a salad, so there's only so much gene editing is going to be able to do. But we are creating a more nutrient-dense salad option to be a substitute for something that's maybe less nutrient-dense. And then now that we've developed our product, gotten good consumer feedback, we're getting our product in the market. We're currently available in retail, we're currently available in food service, so in restaurants and cafeterias. We hope to have our product on retail grocery store shelves soon. In order to do this, we've been engaging with regulatory authorities, probably many who are on this call, to go through the appropriate process for a gene-edited product, and to inform the agencies about our intent to launch these products in the market. We've done this in North America, in the US and Canada, in several countries in South America, and we're beginning to engage agencies in Europe as well as in Asia. Just coming up on my last slide here, I wanted to come back to this value chain and sort of the difference Pairwise sees as our role in all of this relative to companies that have come before us. We are the technology owner. We are developing the product. We are partnering with the value chain in the middle for a high quality product that then gets on to the consumer under our own brand. And through all of this, we have great stewardship and quality and traceability so that we're able to take our product from seed to store and know and tell that whole story. So I'll just close by saying we are really proud of our first branded product in food service and soon in retail, and of the ability to use these great innovative technologies like CRISPR to bring what could have been produced through breeding to the market and to the consumer faster. And we look forward to what Pairwise will be able to show you next. Dave, with that, I think I am finished.
Very good. Thank you very much, Nicole, for that nice talk. Very concise. I think we have time for questions now, and we can also hold them till after Peggy's talk. But does anybody have a burning question now? Just bring yourself off mute. You should be able to come online and ask your question verbally, or you feel free to put it in the chat as well. I don't see anything in the chat right now. maybe a warm-up question for me, Nicole. You talked about a little bit about the specific timelines for the greens coming down towards the end, and it was pretty rapid. But can you, I mean, really great turnaround time, and so thanks to all the agency personnel that were involved in that, and some really good progress there. But could you also talk a little bit about the timeline from discovery to commercialization? I mean, when did Pairwise first start talking about these as these edits as targets, and what was that timeline like from the early discovery discussions all the way to commercialization?
Yeah, it was quite fast, particularly for those of us who have spent a lot of time in the biotech arena. We know product development, plus then the time to actually get a product through the regulatory system can be quite lengthy. Pairwise just turned 5 years old. We were probably a year in before we started working on the mustard green project. So in about 4 years, we were able to go from idea, hey, this would be a great target for editing, to having a product in food service. That seems incredibly quick. And then coupled with the great response we're getting from consumers, from our partners, it's been really quite a whirlwind. We look forward to launching a new generation of greens soon, as well as some other produce products. And again, we've had great engagement with the regulatory agencies in the US and Canada and other parts of the world to really take appropriate systems to review these products and make sure the innovation is able to get out there to the consumer and to support new developers in this space.
Good, thanks, Nicole. So a couple of questions came into the chat. Neil Hoffman says, the greens are available in food service. Are they available in supermarkets yet? And if not, when do you expect them to be available?
Yeah, good question. Yeah, greens are available in food service. They are coming to grocery stores in select markets soon. Check out our website for more information as those get ready to launch.
Good. Brian Irish says, what evolutionary role do glucosinolates have in Brassica jonesea? Are there any drawbacks to eliminating this function in the gene-edited product, i.e. effects in plant production?
Yeah, we understand that there is an enormous amount of literature out there that explains the roles glucosinolates play in plant defense. We also know that through breeding, we have been able to reduce glucosinolates in these varieties. Think about the difference in taste between raw kale or raw collard greens and raw broccoli. There's a definite reduction in pungency, and that often correlates with reduction in type or amount of glucosinolates. We've been able to recreate through editing some of those naturally occurring loss of function mutations in these myrosinase sequences that exist naturally or have been induced through breeding, and recreate them in the varieties that we're interested in working in. So we treat our mustard greens just like you would treat any other mustard green you were producing for produce use. And we haven't seen any difference in that space.
And then Nicole, David Fang, is your product price competitive as compared with regular leafy greens?
Yeah, pricing is interesting. And we have done benchmarking against some of the more premium products and are priced competitively against those.
And then do you see the one from Kaishu? What is the regulatory difference from the US to other countries for approvals of commercial release of gene-edited mustard greens? And I might add to that, what countries are you pursuing commercialization in today?
Yeah, so right now, our commercialization is limited to the United States, where we were able to go through the legacy MI-regulated process very quickly with the USDA. We realized the regulations have modernized in the last few years. We've also been able to take advantage of modernized regulations in Canada. So we appreciate the work Health Canada and CFIA have done in that space, as well as countries in Latin America like Colombia and Honduras. I think it's difficult to compare regulatory systems. There are different triggers, of course, for every regulatory agency, and we've just been working really closely with those authorities to make sure we are able to bring our products to market in the appropriate way. Was there a second part to that question, Dave?
Just other countries that you're specifically pursuing.
Again, right now, limited to the US, but potential market opportunity in Canada, as well as, maybe Australia, Chile. We look at this from both a seed production freedom to operate. Can we produce the seeds we need? And then the produce is typically grown in the regions in California where most produce comes from. So.
And Nicole, Alan Van Deynze asks, will you market stating technology written on label or let the product speak for itself?
Yeah, good question, Alan. I had a slide. I think I took it out. We have been really intentional in our branding and in our marketing to not shy away from the fact that we have produced these products through CRISPR. And something to that effect will be stated on packaging is our intent. It's also on our website. We also intend to put a QR code on our packaging for people who want more information. All of that's still pending as we look to launch our product in the retail grocery stores.
And then Thomas Luberstadt asks, what is the development cost for gene editing versus old style transgenic products?
I'm not sure I'm the best person to comment on the cost, but due to the modernization we've seen the regulatory agencies take, there has definitely been a reduction in that pre-market step in terms of studies taken to clear regulatory systems. And so we do appreciate that. In terms of the development cost, we still face the same challenges a conventionally bred product would in terms of several seasons of selection to make sure you're bringing your VI or your MVP lines to market, as well as just other standard industry steps like processing, specifications, packaging, things like that. So all of that's still the same, but the regulatory burden has definitely been reduced as we modernized for gene editing.
I might just comment on that, having been in the transgenic world since, very early stages of GMOs and corn. I think a lot of the estimates now, above $100 million, 150 million, I've seen as much as, I think, and those are fully loaded costs of everything, regulatory costs, development costs, discovery costs. And it's not hard at all to get to 150 million if you think about a large project costing, an individual cost center budget of $10 million a year. You work on it for 15 years and you've got a sunk cost of 150 million. That's kind of simple math, but that is easily how you come up with these kinds of large numbers. And as Nicole well said, Pairwise has been in business for 5 years. And a lot of the development timelines from discovery all the way to final product approval, 15 years, 12 to 15 years is not an uncommon, flight path for GMOs, even still today. And that timeline hasn't gotten any shorter. It's almost gotten longer. So it's an interesting question and comparison. There's a question on glucosinolates have anti-cancer properties. Is it a good thing to reduce it?
Well, we know that we already consume products that are low in glucosinolates, or lower in glucosinolates. And so we believe that our lower pungency mustard greens would be comparable to those products that had been produced through traditional breeding to also have reduced pungency.
Mm-hmm. And maybe let's end on this one and keep moving. Nicole, there's a question about, do you see off-target effects in edited products?
Yeah, as part of our edited line selection, we follow industry standards for how mustard greens should be evaluated in the field. And in doing that, we did not see any performance differences from our edited material to what we were comparing to conventionally.
Very good. Okay, well, thank you, Nicole. And there's a couple more questions in the chat. And I'd say we have two options. If you like, Nicole, you can answer them while Peggy is speaking. You can answer them in the chat, or we can hold them to, yeah, if you have further commentary you'd rather verbalize, we can hold them till the end during when we have the panel discussion. So either way should work.
Yeah,
Okay.
thanks, Dave. Maybe we hold
Okay.
them till the end to make sure we're focused on the story Peggy's going to share with us.
Yep, very good. Okay, thank you,
you,
Nicole, very much. Appreciate it. All right, Peggy, are you ready to go?
Yes, I think so. I'll share my screen. Okay. Are you able to see that?
Yes, perfect.
Okay. Well, good morning, everyone. I'm pleased to have this opportunity to share some of our work on the potential to mitigate peanut allergenicity. My name is Peggy Ozias-Akins. I'm a professor with the University of Georgia, also a faculty member in the Institute of Plant Breeding Genetics and Genomics. I was director of the Institute for 10 years. I also did speak at the first symposium that you had back in February of 2020, and that was also on a peanut-related topic. It was on aflatoxin rather than allergens. So just to put the importance of peanut into context, it is grown across the world in tropical and subtropical regions. China and India are the largest producers of peanut, and it's primarily used there as an oilseed crop. The U.S. is also an important player here, although we're typically fourth or fifth, depending on the year in production. But the U.S. does have some of the highest yields as well as highest quality peanuts. And much of the peanut is in the U.S. is used for confectionery purposes. So within the U.S., the peanut is largely grown in the Southeast. However, my home state of Georgia produces about 50% of the U.S. peanuts. So you can see why there's a lot of research conducted in Georgia. And I'm going to be talking primarily today about not a product that we're intending to bring to market, at least at this point. So nothing like conscious greens, but really more about some upstream research that we've been doing. So peanuts are a really interesting crop biologically in that it flowers above ground, but it produces its fruit underground. And so peanut is typically self-pollinating. It is a legume. You can see that there is a bee visiting the flower, and we do see this in the field, which leads to some level of outcrossing, normally no more than around 10%. But after the flower is pollinated and fertilized, the seed begins to grow, but then it actually undergoes a quiescent period. And there is a region around the tip here of what we call the peg, where the ovary is located, and that gets pushed underground. And so it senses when it is underground, and therefore the seed then resumes growth, forming the seeds within this pod. So normally the pod will contain 2 to 4 seeds. The type of peanuts that we grow mostly in Georgia are called runner type peanuts, and they are typically 2-seeded peanuts. And a large part of our crop is used for peanut butter. So it also has been speculated that since the pods are produced underground, they're exposed, of course, to many soil-borne pathogens, and perhaps some of the proteins that are produced have evolved some functions that are for crop protection. Peanut evolved in South America. It is a polyploid crop. It's an allotetraploid, so similar to the brassicas, as many of our crops. It evolved from hybridization of 2 diploid progenitors. Both of these progenitors are still extant, Ericus duranensis and Ericus epaensis. We call the duranensis the A genome progenitor and epaensis the B genome progenitor. There actually is only one accession of epaensis known, and therefore there's no diversity to work with from epaensis. However, with duranensis, there is some diversity available. So the hybrid was naturally occurring. It did double in chromosome number to produce the allotetraploid peanut, is inbred and homozygous. But then what this did lead to was genetic isolation from its diploid relatives. And it likely, the hybridization and polyploidization event likely occurred very few times and perhaps only once, leading to a severe genetic bottleneck and therefore reduced diversity within the cultivated peanut. And of course, if we need resistance to biotic and abiotic stresses, as we do in improving crops, then often that level of resistance that we need is not available within the cultivated germplasm. We do sometimes find it in the wild germplasm, but then of course we run into the issue with the cross-compatibility with the diploid species. And in peanut breeding, breeders are focused largely on fungal and viral diseases. I've listed several fungal diseases here, such as leaf spots, rust, white mold or stem rot, and then also the infection by Aspergillus flavus, which can lead to aflatoxin production, particularly under environmental conditions where we have elevated temperatures and water deficit. Tomato spotted wilt virus is also a serious problem for us here in the southeastern US. And there really is no chemical control for the viral disease. It has to be controlled through genetics. So one of the traits that breeders really have not been focused on is peanut allergens, or reducing allergens, or looking for allergens that are less prevalent. But we do know that peanut allergy is a problem. It does affect approximately 1% of the US population. It does persist into adulthood, certainly more than allergies to some of the other big 8 crops. And although rare, we know that fatal anaphylactic reactions can occur to peanut. And so there has been some interest in looking at the potential to reduce allergens in peanut. This is a big challenge because there have been 18 allergenic proteins identified in peanut seed and recognized by the International Union of Immunological Societies. And therefore, I don't think it's feasible to produce a non-allergenic peanut. Some of those proteins are essential proteins to growth and development, and then some of the ones I'm showing here are actually seed storage proteins and therefore vital to the seed germination and growth. These seed storage proteins, the majority of them are considered major allergens because they are recognized by greater than 50% of peanut allergic individuals. These seed proteins 1, 2, 3, 6, and 7, ARA-H1, 2, 3, 6, and 7, also comprise the bulk of the protein in the peanut seed. So ARA-H1 is a globulin called visilin, can be up to 23% of the total protein in the seed. Glycinin, or ARA-H3, is the most abundant, being present at up to 83%. And then the conglutins are the least abundant, and that includes ARA-H2, 6, and 7, and those can be present at up to 18%. So as far as their allergenicity, the conglutins, and specifically ARA-H2 and 6, which have been tested, are the most potent allergens. In fact, they're 100 to 1,000-fold more potent than ARA-H1 and 3. They are recognized typically by up to 90% of peanut allergic patients. And it's also been shown that their allergenic potency can be increased upon roasting. And roasting is one of the primary processing methods that we use for peanut in the US prior to the production of peanut butter or use of peanuts in confectionery products. So we focus more on the conglutins, also known as 2S-albumins. And of course, the number of genes that are underlying the production of these proteins is complicated by the polyploidy of peanut. ARA-H2 is one of the predominant albumins, and it has 2 isoforms. These 2 isoforms are produced by 2 genes, one in the A and one in the B subgenome of cultivated peanut. And they're very similar in their nucleotide sequence identity. One of the main differences between the 2 is that the B genome copy has 36 nucleotide insertion that is encoding 12 amino acid insertion compared with the A subgenome copy. So it's a little more complicated with ARA-H6 in that we have 3 genes. We have 1 in the A subgenome and 2 in the B subgenome. So there's been an extra duplication in the B subgenome for ARA-H6. Between these 3 genes, there is 92% sequence identity. But if we then compare ARA-H2 and 6, there's only 63% nucleotide sequence identity. The duplications, or all 3 of these genes in this gene family, ARA-H2, 6, and 7, are also found in tight linkage on 1 chromosome in each subgenome. So this is just illustrating where the major difference is in the nucleotide sequence when the A and the B subgenome copies of ARA-H2 are compared. It is this insertion. And this insertion is probably has some importance because it is translated, and it's translated such that there is an immunodominant epitope, DPY-SPS, that actually now becomes repeated 3 times in this copy. So there is DPY-SPS here, there's DPY-SPS here, there's also DPY-SPS here. So this is an immunodominant epitope, meaning it's recognized by a large percentage of the IgE from peanut allergic patients. Again, the similarity between ARA-H2 and 6 is not as great, and these black boxes show where that similarity is located. And so this does have then implications when we start to consider the design of guide RNAs for genome editing. So my lab has been involved over close to 20 years in exploring reverse genetic approaches for reducing peanut allergens, and primarily focusing on the congluteins, initially primarily on ARA-H2 because that was the most abundant congluten, and because now it's also known that those congluten proteins are the more potent allergens. And so initially we were taking a transgenic approach with post-transcriptional gene silencing, also known as RNA interference, where we took a portion of the ARA-H2 gene, introduced it as an inverted repeat in a construct, and then saw that it could actually reduce the expression of ARA-H2 as well as ARA-H6, which I'll show in a moment. We've also explored other reverse genetic approaches using potentially non-transgenic means. One is random mutation. The random mutation, then we have to actually search for those particular mutations in the genes of interest. But now more recently, the targeted mutation using CRISPR-Cas9. So with the knockdown or the RNA interference approach, we were successful in generating transgenic peanut now that did reduce the expression of both ARA-H2 and ARA-H6. And we looked at this at the level of the protein using antibodies that were specific for the particular allergens shown here, which we could detect through a Western blot, gel electrophoresis, and then Western blot with those specific antibodies. We could see then clearly that there was in the transgenic line shown in this particular gel or Western blot, we could see the reduction in ARA-H2 and ARA-H6, but no reduction in the non-targeted allergens, ARA-H1 and 3, or at least not a quantifiable reduction in the protein gel. We also then looked at using a more quantitative method of liquid chromatography, mass spec analysis, and spectral counting to, again, substantiate that there was a significant reduction in ARA-H2, but also ARA-H6. And then we saw a small reduction in some of the other seed proteins. We, in addition, saw an increase in some of the other seed proteins, and 2 of those being other allergens, ARA-H7, which is another congluten, and ARA-H8, which is a pathogenesis-related protein. So it's been demonstrated with other studies in manipulating seed storage proteins that if one is reduced, it's likely going to be compensated for by an increase in other proteins. So another question we had was really, how does the peanut seed itself tolerate the elimination of a seed storage protein? Even though we knew that there was some compensation, we didn't know if there was a specific function of
Morning, Shannon.
these major conglutens that we might have to be concerned about. And so we looked at different traits in order to try to determine this. ARA-H2 had been shown to have some trypsin inhibitor activity and therefore potentially some antifungal activity. And so since we were working with aspergillus and aflatoxin reduction, we wanted to test whether elimination or severe reduction of ARA-H2 had any impact on aspergillus growth. And in fact, we didn't see any significant difference between the transgenic and the non-transgenic lines. Similarly, we didn't see any significant difference between non-transgenic and transgenic lines in terms of seed size or root elongation shortly after germination. So it appears that seed vigor would not be seriously impacted by the reduction in these conglutens. However, the question that is perhaps more important is whether reducing these allergens is actually going to reduce allergenicity. And it's a difficult question to answer because really to get a definitive answer, you need to do challenge with humans, but that's not really feasible. Certainly not feasible for us because we don't work in that space. But we have worked with collaborators to gather some evidence of the potential for reduction in allergenicity. And this is just simply showing that there is reduced human IgE binding in some of the conglutens in these transgenic lines. And this was consistent in one transgenic line shown here, the 11.1.1, with 3 different patient IgE sera. And so there's essentially no binding that was detected, at least at this level of detection on the Western blot. But we used another couple of assays in collaboration with different people. This is with Manel Jordana at McMaster University quite a few years ago now, where he has developed a mouse model of anaphylaxis. And we were testing a transgenic line compared with a non-transgenic for whether there was an alteration of allergic reaction in this in vivo model. A 90% reduction in ARA-H2 is not sufficient to alter that allergic reaction. And that's probably consistent with what is known now about the potency of ARA-H2. But in another assay that was conducted in collaboration with Stephen Dreskin at the University of Colorado, with this particular transgenic line and using an in vitro assay with rat basophil cells, which were sensitized with human IgE and then challenged with crude protein extract from wild type and transgenic lines, it actually showed that the histamine release was reduced by challenge with the transgenic line compared with the wild type. So there certainly is the potential to reduce the allergenicity, but these are knockdowns, and so it's not complete elimination of the protein, and therefore it's probably not a route that we would want to take, particularly for a potent allergen like the conglutens. So therefore that led us into the mutation strategy. And initially when we started this, CRISPR-Cas9 or CRISPR had not even been discovered. And so we were focused on generating mutants through random mutagenesis with chemical treatment. And this is one of the typical chemicals that's used is ethyl methane sulfonate. So as far as the mutagenesis part, it's nothing that hasn't been done for decades in breeding, in mutation breeding. What was different was just the method of screening. So since at this point we did have gene sequence of the allergen genes and specifically ARA-H2, The method that was used was to screen for changes in the gene sequence using tilling, which is an acronym for targeting induced local lesions in genomes. And tilling can be used not only with induced mutations, but also looking for natural mutations. And so we did extend this to what is called eco-tilling by looking at Erichis durinensis. As I mentioned, there is variation in Erichis durinensis. We knew that there was genetic variation. We had access to a number of different accessions that had been collected. And so we looked at the what we call the ERA D2 now, which is the homologue of ERA H2 in durinensis, and did find that there was variation in sequence in this progenitor genome. And this is just an example where one of the accessions did have a change that also changed the amino acid sequence. And you can see that it changed the amino acid sequence in this immunodominant epitope, DPY-SPS to DPY-TPS, and that actually reduced the IgE binding, at least from this patient, to very little. It was a little bit more from this patient. So this change, which does occur in nature, actually did significantly reduce the level of IgE binding. With the induced mutation strategy, we screened really for ARH2 induced mutations in both the A and the B genome copies, finding a number of mutations, some of them silent, some of them that did generate amino acid changes, but those changes were not predicted to have any function. And then one in particular that was predicted to have a function and actually to be a knockout because it disrupted the start codon, or the ATG, at the beginning of the gene. And this was in the B subgenome copy. When we then looked at the protein that was generated by this mutant line, we could clearly see that the protein from that B subgenome copy was eliminated. It was no longer present compared with the wild type. We could see that both on this one-dimensional Western blot, but then also on this two-dimensional gel, where due to post-translational processing, there are actually 4 spots. And we could see that in the B genome copy of ARA-H2, the spots only represented the wild type and not the mutant line. Whereas in the A genome copy, we had protein from both the mutant as well as the wild type that we could detect. So with the advent of the CRISPR technology, the obvious route was to try to knock out all of these congluten genes. We knew that now there were 7 genes present in the cultivated peanut genome, 2 of ARA-H2, 3 of ARA-H6, and 2 of ARA-H7. As I mentioned, these are all linked, and therefore, with a random mutagenesis strategy, the probability of actually getting mutation in all of these genes, or even then being able to, say, recombine, if we were able to get mutations in different lines of 1 or more copies, it would still be very difficult to combine them through breeding because of their tight linkage and the lack of likely recombination between those copies. So using the CRISPR-Cas9, we wanted to try to knock out all 7 genes. Due to the low similarity between ARA-H2 and 6 or 2 and 7, we were not able to design guide RNAs that would target all 7 genes, but we were able within each group, so within ARA-H2, within ARA-H6, or within ARA-H7, to design common guides. And we decided to hedge our bet by actually designing 2 guides for each of those sets. These 6 guides were then put together in a polycistronic construct along with the nuclease Cas9 and introduced into peanut. And so initially, of course, the peanut was transgenic. And we have now advanced these lines to the T2 generation, where we have in some cases looked at T1 and others looked at T2 seeds for the production of these allergenic proteins, so the conglutens. And these two are Western blots, one with anti-ARH2 antibody and another with anti-ARH6. You can see that ARH2 is the predominant congluten and very abundant in the wild type. And one of the gene edited lines, we can see that we have eliminated the B genome copy of ARH2 or eliminated the production of this protein, but the A genome is still produced. However, in some of the other edited lines, we now have apparently no ARH2. And based on our sequence data, this is what we would have predicted. ARH6 is not quite so clear, but you can see that there certainly is a reduction in some of the edited lines for ARH6, and our genome sequence does suggest that that should be the case. We do not have a specific antibody for ARH7, so we haven't been able to separate ARH6 and ARH7 or determine whether we have cross-reactivity there at this point. But it's likely that we are able to knock out all 7 genes if we put in the effort. So to summarize, there are many allergens in peanut seed. I mentioned that there are 18 that have been recognized, but the conglutens are the most potent. And so those are the ones that we have focused on in our research. The knockdown does not appear to compromise seed quality. We do not have sufficient data from the knockouts to know if that's also the case. We do know that knockout is possible through mutation, either through random mutagenesis. It would be very difficult to combine mutations in all 7 copies of the congluten genes. And therefore, it would require knockout using the CRISPR-Cas9 multiplex targeting. And we have shown that that should be possible. And maybe we have already done it. We just don't have all of the evidence to confirm that at this point. So just to acknowledge a lot of the people who have been involved, I mentioned the investigative groups at McMaster University and University of Colorado, also collaborations with USDA ARS here in Tifton, as well as in New Orleans, University of Missouri for some of the proteomics work, and then a number of postdocs and technicians that have worked on the project in my lab over the years, plus all of our sponsors. So thank you for your attention. I'd be happy to address any questions.
Very nice talk, Peggy. Thank you so much for that. I'm gonna pull an audible here since we have kind of a dynamic schedule this morning, and we have a little bit more time. And so we're gonna hold questions for Peggy until the end, because I'm going to insert one more topic here. And that is Alan Van Deynze is with us this morning and this afternoon. He'll be your moderator. And he's been working on talks over time, and he's the director of Seed Biotechnology Center at UC Davis. And he's been working on this talk and has given it before, and hopefully to none of you, on the Evolving Breeders Toolbox. So we're going to take another 20 minutes because I think it fits squarely into the topic for today. And since we have a little bit more time this morning, Alan's graciously offered to run through his presentation, and then we'll have a panel at the end of this in about 20 minutes. And the rest of the schedule for the rest of the day remains the same. So for the afternoon session, which will start at 12:30 Eastern time. So Alan, with that, can you go ahead and go through your presentation?
Yeah, can you hear me? Let me see, I can get my video going. Yeah,
Very
can you hear
good.
me now?
Yep, hear you
Very
good.
good. Yeah, thanks a lot for inserting me and things like that. And like I say, I've modified a little bit of the talk, but this is a talk that I put together for the ISBR meeting and global policy back in St. Louis, but I thought it'd be pretty relevant. And like I say, hopefully not everybody in the audience has seen it already. And this is a lot of the work that I've been working on, and that's what's going on. I just wanted to kind of just give an idea of sort of really, what the title says, the gene editing. A lot of people talk about CRISPR, and is very well known, I'd say, even in the public a lot of times. but it's such an evolving tool. Gene editing is such an evolving tool, and I want to talk about some of the challenges and things that are going on. And I've been at, I've had the pleasure of also participating, like Peggy, to to these meetings, and I'm a pretty active member on the National Association of Plant Breeders as well. And I give some thought, and people, I get pushback on this this definition of plant breeding.
Hey, Alan, excuse me, we're seeing your whole slide deck, not presentation.
Oh, sorry about that. Let's see. I'll stop share and try again. Let's see. Share screen. This one will work. How about now?
Yep, perfect. Thank you.
Okay, great. And so I like to give a definition of plant breeding up front, and this is like my definition. There's many definitions. Plant breeding is really not a science. It's more like it's really a discipline of sciences, and it's rooted on population genetics, selection theory, quantitative genetic, and statistics. So we like a lot of numbers. And it's for crop improvement and increases number of support technologies. And the list, keeps evolving. And I'll give you a list of some of the technologies that we work with. And of course, gene editing, that what we're talking about today is just one of those technologies. And conceptually, it's simple. We generate, genetic diversity, usually by controlled crosses. We impose selection to fix traits so that we can reproduce them. But an important component is that there's a lot of testing going on over multiple field locations, over multiple generations and years, really to make sure that they work in the farmers' fields and they have good consumer appeal, processor appeal, and really stakeholder appeal, for the targeted markets. So then we put out the best varieties, and then those varieties become parents really for the next cycle to keep improving, because there has never been the perfect variety. And when anybody has said they have the perfect variety, there's a new disease that comes up and said, nope, we've got you. Or a new insect comes up and it says, no, you're not so perfect. And so we keep going. So that's good for us. Let's see. So where do GMOs, the traditional transgenics, fall into this? They have typically been a source of unique genetic diversity. And I like to use the word transgenics, really, where trans means from another species. Usually that was the original idea, like BT going into crops, as opposed to, but GMOs have been really lumped where when we add a gene from the same crop, when we insert a gene from the same crop, they've been treated the same on a regulatory basis. Gene editing, we have a lot, we're seeing, some different regulations, which we're very excited about, especially as public breeders. They also are sources of new genetic diversity, but what we hope to do, the efficiency of gene editing, we hope to make more genotype independent. The GMOs have been limited to things that agro-transform. That's usually limited to a variety within a crop that you can do. There's not many crops, except for maybe tomato and brassicas, that you can actually transform really easily on a routine basis. So if we can get transformation or gene editing, even without transformation or culture, it moves to the really adding a new trait to an already, an elite line that's already commercial. So it's more like a backcross system that could be a lot more efficient, actually, where we still, put it in the farmers' fields and test it and put out the variety. So gene editing fits in the cycle perhaps in a different place than traditional GMOs, and largely due to its efficiency. I was fortunate. My first job was with a small company called Calgene in Davis. I was the breeder for transgenic oils, and we had this oil where I would say GMO killed it. Unfortunately, the market killed it, where the best market for it was really confectionery coatings, and the best market was in South and Central America for this. What we did is we added a gene from the bay leaf. This 12 lauric acid is a fatty acid that's has a short chain fatty acid. There's 0 of it in canola. We were able to get up to 47% in canola, and this had canola quality. We sold it really as Safola margarine, so it was actually a sustainable, sold as a sustainable oils, low pesticide, but it actually was trans fat free. And so, but we were 10 years ahead of the label. We didn't know trans fats were actually worse than saturates, fatty acids for you. And part of that was timing. But this came out, was fully registered. It was a GMO. And we inserted actually 3 copies of genes for lauric acid from the bay laurel trees, which were selected really just south of Davis here. It was fully approved by USDA, by FDA back in 1995 in Canada and the United States. And we actually grew 100 acres of seed in the UK, in France, and Germany. We had 100,000 acres growing in the United States and Canada back in about 97. But really GMO regulation and pushback is what killed it. So Roundup Ready was not really the first things out. This was out about the same time as Roundup Ready. Jump to 2022, and I'm jumping in a little bit to the talk that didn't quite make it today. Professor Kathy Martin out of John Innes Center of the UK created this high anthocyanin in tomato. So people might say, well, we've seen purple tomatoes before, but if the purple tomatoes that were on the market were from a wild cross and only the skin really was purple, and it was only purple on the side that was exposed to the sun. The bottom of the tomatoes were green a lot of times, but the internal side, basically the pulp and the mesophyll of the tomato, was actually red on that. And actually, to be honest, these wild tomatoes are getting better now, but a lot of them didn't taste so great because they were a wild cross. So they had to really breed all the flavor and production traits and everything back into it. From back crossing or things like that, where it's a 2-gene GMO insert. It's not gene editing from Snapdragon, and it's a promoter that basically allowed the anthocyanin to be produced throughout the tomato. I am very fortunate that this company, Norfolk Health Produce, is actually in Davis right now, and I'm growing these in my garden. I've been eating them for about the last month. They are really a delicious, sweet, great product. So I have good hope that this will be a very successful product, like we've seen some Pairwise this morning as well. So I don't need to tell most people on this, but I call this traditional gene editing technologies. This is the original, say, transgenic technologies, and some of the aspects, limitations about it. Agrobacterium makes it very efficient. We hijack agrobacterium. We disarm it so it doesn't give gulls, which is what it does in dicots naturally. It is really a pathogen, but it's disarmed. So we use it and its functionality to integrate DNA into the chromosome, really somewhat randomly into the genome. And that's why it's inefficient. You have to have the right place in the genome, the right promoter, and so on for things to work out. And give you an idea, maybe like GM canola, the first stuff that came out, they had 1,000 events for Roundup Ready canola to get 1 or 2 that worked. For the lauric canola, we did about 120 events for it, to get something that we liked. In gene editing, it's so much more. You basically need 10 plants usually, and you have 5 of them that give you what you like. So chromosome integration, which is what we want, limited hosts and genotypes, and that's because it requires plant regeneration or efficient plant regeneration and transformation. And so that's a limitation for most crops. Or we can use a gene gun, which is a shotgun approach. Literally, it's actually a 22 approach. Initially, it was a 22 bullet. Now we just use an air gun essentially to blast DNA into the genome. It may expand the genotypes you can do. It requires plant regeneration, but you do get gene fragments and multiple insertions of the gene that you want. So that's, the good part is we may be able to expand crops or genotypes. The bad part is it's a little bit messier when we get in there. So we have to do a little back crossing to clean that up. CRISPR, just to go on, chromosomal integration is actually optional. There are CRISPR technologies or gene edited technologies, which I'll explain that the gene edit itself doesn't require CRISPR to integrate into the chromosome. It really just needs to be, the protein just needs to be expressed in the cell. So potentially they can expand the host and genotype. So we're excited about working on that. And it may, and we're seeing it may be more acceptable to regulatory and consumers. So CRISPR, this is CRISPR per se Cas9. It does a double-stranded cut. We take advantage of DNA repair system that is imperfect, that we get small deletions, sometimes larger deletions if we engineer it, or we can have insertions and, to do more precise gene editing. Gene editing so I'm going to go through sort of systems that a lot of people are working on to try to make this more useful in many crops. And we see this, gene edited is exploding both in academia and small companies, as opposed to really sort of the big five and, 2 or 3 big companies that on crops that have large acres, for GMO. And that is partially because the investment, and David mentioned, the cost of investment for traditional GMOs is quite expensive. Protoplasts are exciting in that it's quite efficient, but the hosts are quite limited to a select few species in Solanaceae, Poaceae, and Brassica families. So it's basically we digest the cell wall, and we were able to basically transfect DNA. So DNA-free is a bad word. There's no integration necessarily of CRISPR-Cas9 or your gene edit tool into the genome of the chromosome, but you actually get the gene edit. All right, so it can be quite efficient. It says so it's transfection, so there's no, like I mentioned, no direct. So transient transfection certainly has different regulatory issues, if any, as far as being exempt, because there is no foreign DNA in the final product necessarily. The entire regenerant plant is derived from the single protoplast. Anytime you hit culture, that this is a challenge with all culture things, you can get what's called somoclonal variants. So just being in culture itself is a stress on the plant, and you do get mutations in the genome or sometimes some off types. You see stunted growth, some abnormal plants, but through breeding, we can backcross those pretty quickly and get, favorable genotypes, and test them in the field and so on. One of my colleagues that I've been working at the Integrative Genomics Institute at UC Berkeley, this is Jennifer Doudna's Institute at Berkeley, a really exciting, I think, graduate student that's just finished up, Evan Groover, that I work with. He did 6,400 single cell gene edits using protoplasts in sorghum. So sorghum actually isn't that good at protoplast regeneration, but they're doing this to study promoters and which parts of the promoters are most efficient for gene editing to try to understand gene regulation. But he did this in 6 months, just to give you an idea of the efficiency. 6,400 gene edits. To me, that blew my mind when he showed me this. There's challenges, for gene editing. I unfortunately my favorite crop is pepper. Pepper is recalcitrant to not regeneration, but through agrotransformation. So we can get plants to regenerate, but they don't transform. So there's a gene silencing system going on there, or really, I think, a hypersensitive response to certain agrobacterium strains. Everybody uses the same agrobacterium strain in general, called EHA105, but there are many other strains out there that are more efficient or less efficient, depending on the strain. We're seeing this in pistachio. We're seeing this in walnut. This is something we're going to try. We're trying in other species as well to see if agrobacterium strains are more efficient, say in crops like pepper, which are really difficult to use agrobacterium in. So we'd like to use agro because it gives you nice insertions and it's predictable and so on. But with pepper, it's just not a thing with the EH105. Other methods to get, for gene editing are basically RNA viruses. This came out of a group out of Ellison, and there's a group at Davis was involved in this as well. So this is tissue culture free. So that's what's exciting. Remember, I talked about the somoclonal variants, and tissue culture is also a lot of work. sterility, you need fume hoods. It takes 6 months, a year to get plants, and you still have this somoclonal variance. And there's a direct relationship really between the time in tissue culture and the amount of somoclonal variants you get. So there's RNA viruses that are used to target, to integrate really systemically, but this is chimeric. You can kind of see these leaves, this marker. This is a marker where they knocked out phytoene synthase no, I'm sorry, PDS, phytoene desaturase, which gives you these basically albino type little chimerics. But in the next generation, you can basically clone these areas, and you have gene edits and so on. So quite efficient. They are specific right now there is a lot of work to get these to work, to be honest, but once you get them to work, they work well. So a different way to get, besides agro, to get things in. Carbon microdots. This is out of another publication came out in 2020. So they're little carbon nanotubes, if you want to think about it. They're little dots that are small enough to get through basically the plasma membrane and into cells. And these dots are charged so that they can attract DNA. The problem with microdots is the micro part. They can only carry a small DNA load. So like small RNAs work, 20-25 base pairs, but you can't get, whole gene constructs on there and things like that. But they're excellent for knocking out genes. We know siRNAs do that. They silence genes or families of genes. So this is an example of using nanotechnology to get basically gene edits or basically gene modifications into the cell and hopefully expressed, expressed into the cell in gene edits. So again, this is not necessary. This is transfection. You're not integrating into the genome. So this is basically non-GM per se, but it's a gene edit. So another, carrier method to get things in. Methods to try to enhance agrobacterium efficiency is to look at basically growth factors that enhance regeneration or speed to regeneration. This is a group out of UC Davis that came out. Juan DeBernardi published this a while back, and he did this in wheat, so a tough crop. This is a polyploid. He did this initially in tetraploid wheat, so it has two genome copies from two different progenitor species, but also works in hexaploid wheat. It works in pepper, it works in lettuce, it works in citrus. And what it does, basically, this is the control where you're getting, say, one plant to, regenerate a shoot. Here you see out of these, 20-odd little callus pieces, whereas here you're getting half of them or more, even 60-70% of the plants are getting shoots quite efficiently, and they can get them quicker, but they also have expanded the genotype range that they could use as well. So this has allowed agro to be used. It's kind of breaking that old tradition. Is like, well, I can transform with agro, but only one genotype, and it's usually a bad one as far as the farmers or as far as a variety is concerned. And so there's a number of these growth factors that are used as gene constructs to help agro do its job. And this is really the impact. Baby boom is another gene that people are using. And so if you have to do traditional agro, you have to do selection in the media through different shooting phases. Then you put you cut out the shoots, you put them, the callus phase, shoots, rooting. And this takes 6 months to a year, depending on your crop system, to get these plants. And without, if you can skip that with these kind of baby boom and Wuschel and GRF type growth regulators, you can kind of skip these steps in the middle. And the shoot induction and root induction, and really get things in a couple months. Say in tomato, in 2 months this works, or in wheat in 3-4 months you've got plants. And much more at a higher rate as well. So this is an exciting way to get gene editing out there. My colleague that I've been working with quite a bit for the next 4 years. This is Marquita Landry out of UC Berkeley. Is working also on carbon nanotubes. So these This is like the microdots, except think of they're cylinders as opposed to the microdots. The idea is the nanotubes that are cylinders have a bigger payload, right? The DNA can now be wound all the way around this cylinder as opposed to just, a little dot. So we can now put bigger gene constructs, multiple gene targeting, for gene editing out there. And again, they're about the size of a microdot in their diameter, so they can kind of get through that plasma membrane into the nucleus. And the idea is then deliver their payload as well and get basically non-transgenic gene edits. The idea and hope behind this is this is work out of my lab. At first we started with leaves, and then we realized I was like, why do we want to do leaves? Leaves don't give you plants, meristems do. So we start using this work on meristems. So these are isolated pepper embryos, and we can get good expression, say, of GFP. In pepper embryos, we can get good expression of GFP on axillary buds, and then also even a little bit less efficient, but also to pollen. So the idea with pollen is not a meristem, but if we can gene edit pollen, they're haploid, we can then use that pollen to cross really any plant we want and then transfer that trait very quickly. So that's our goal. This is all on pepper. And the idea is that we can, make things this is a non-agro based system. The idea is to have a transfection system, and that we can be genotype independent. And is it going to be chimeric? Likely, but we can select, sectors. No regeneration if we can hit meristems. No regeneration required, and limited or no somoclonal variants because there's no tissue culture. So that's our goal. I would say right now the jury's still out. We're trying to do the gene edit step. We can express Cas9 at a very high level in the cell right now, in these tissues. In embryos, we've only tried, and so we're doing the experiments right now to look, can we actually get good efficient gene editing as well? So we're pretty excited about that. Just to expand what's out there, and this is old news. This is a publication out of Collier, out of UC Berkeley, or USDA and UC Berkeley in 2018. So this work was really done in 2014. This is called the agrobacteria system for gene stacking, and this is in Arabidopsis. And the group was able to systematically stack 25 genes and express those genes in Arabidopsis. I mean, so we're not talking single gene knockouts. We're talking multiple gene insertions at a single targeted spot in the genome. This is a breeder's dream. If I can have, my favorite three disease resistance genes, abiotic stress genes, quality genes, all in one locus, that segregates, 1-2 to 1 as a single Mendelian factor. So in an F2 with 99% certainty, I can get one plant with all those genes with 16 plants, but with five genes, I need about 3,000 to get with just 5 genes, 3,000 plants to get, the hope of get all the genes together and things like that. So this is a breeder's dream. This is, people are doing multiple gene edits. You've already seen that. In our first talk, targeted 17 possible genes, for example. So we really need to be thinking of multiple genes, multiple gene edits, and this is it's essential, really. Even in diploids, there's very few cases where you knock out a single gene, and that gives you the work. I mean, it gives you the trait you want, or do a gene edit on a single gene, because most plants are ancient polyploids, and most gene families or most genes have families. And it's rare that you can get a single gene edit. So I think the regulatory system really needs to look at that in their exemptions and things. So finishing up, innovation. This slide I put together a while back as well. This is not things that plant breeding did for you. These are things that enabled big jumps, jump to that next plateau, if you want to think about in plant breeding. So plant breeding per se really started with understanding of genetics, good experimental design, and look at the timelines. We stand on all these people's shoulders. Biotechnology came out really in the 50s. When you think about tissue culture, mutation breeding, transgenic, in 1980 or so. Quick fact: next month there's going to be a celebration for for Dr. Montague, who was one of the original founders of biotechnology. He's 90 years old, so that's going to be next month in university again. So I hope to make that birthday. So he's been doing it for a while as well. But speed to market technologies, this is something Norman Borlaug took advantage of, different nurseries for stability, but doubled haploids, which we're going to talk about later this afternoon, machine learning, artificial intelligence, and so on. And then, of course, good regulation, intellectual property are key innovations in plant breeding. And just some acknowledgements. This is he goes by Trent. Trent has done all the work on carbon nanotubes myself. Teresa keeps my program running when I'm doing things like this. And then, of course, Marquita and Dr. Luca Comai, who's helped a lot in my work. Okay. Thank you.
Very good. Thank you, Alan, for filling in there. And I think that topic was very appropriate for the rest of the subject matter today and fits right into the discussion. So I think all three speakers this morning really captured the complexity of genetic variation that we have, and genome editing and other tools to be able to harness that variation and leverage it for plant improvement, plant performance improvement. So let's have Peggy and Nicole come back on as well, and I'll start going through the chat. And so
address the question. Oops, somebody needs to put their microphone out. Please. Yeah. So our first speaker is going to be Jeff Habben. And Jeff is a senior research manager and laureate at Corteva Agriscience. And he's going to talk to us about multiple gene editing or multiple disease locus in maize. So with that, Jeff, I think if you can share your screen, we can get started. So yeah, reminder everybody, please put out, take your microphone, get muted for now.
So you can see that full screen, Alan?
Yes, we can.
Okay. So thanks to Alan and thanks to Dave for allowing me to talk about some of the gene edited work that we're doing here at Corteva. So maybe I should just pause here, and for people that don't know, that Corteva Agriscience is a merger between the Heritage Pioneer Hybrid Company and Dow AgriSciences. And so the seed part is located in Johnston, Iowa, and the chemical part is in Indianapolis. And so I lead several different teams here in Johnston around seeds. So disease resistance is what I'm going to talk about today, but we're also using genome edited and agronomic traits in our core crops of soybean and corn, and also very actively using genome editing to alter seed composition, primarily in protein and oil crops. Okay, so creation of a genome edited disease superlocus. So what are we talking about here? So I'll refer to it probably as DSL a lot as an abbreviation. So this is a multiple disease resistant package of native genes that we're putting into a single locus in corn. So I'll just repeat that. So looking and identifying multiple disease resistance genes and essentially moving them from corn into elite corn at a single location. And so before I start and dive into the maize DSL, I want to give you a little bit of background about genome editing at Corteva. So Corteva is using genome editing to discover and develop multiple product concepts, and this is across several different trade areas. So these are shown on the left here. Disease resistance, what I'm going to talk about today, a lot of it targeted towards foliar diseases in many different crops. Enhancing feed and oil, primarily in protein and oil crops, and this is to decrease anti-nutrients, to increase protein content, to increase digestibility, are just three examples. Farm productivity is looking at agronomic traits, primarily to increase yield and or yield stability. And this is in both corn and in soybean. And then active areas around smallholder farmer solutions. And so 2 great examples that we have are, number 1 is using genome edited to reduce problems from striga and its infection of sorghum. And this is targeted towards Africa. And a second great example is MLN. So maize lethal necrosis virus is a nasty disease in Africa again. And we have a collaboration with Simmet and Gates Foundation to create genome edited approaches there. And we've had some really great success there. So those are examples of different traits that we're looking at. From an overall standpoint, we're positioned well to harness natural variation, looking for native genes and using genome editing to both from a discovery and product delivery standpoint. So 4 areas there are, number 1, access to CRISPR-Cas systems. So we have a lot of interactions not only within the organization, but outside the organization to improve efficiencies as far as CRISPR-Cas is concerned. Elite germplasm in major crops. What this allows us to do is to do discovery research directly into our elite germplasm. And then that can roll right over into product concepts and a product pipeline if we feel that we have enough efficacy. The third bullet point there is deep genomics knowledge. So I think one thing that kind of goes underappreciated in this CRISPR area is the genomics technology, how much that's advanced and how much it plays such a role in designing our targets, designing the edits, looking for any offsite problems that may occur. It's really helped genome editing advance, and we have some of the best tools and people that really help our CRISPR-Cas effort. And then final one there is advanced genomic editing methods. And so this really helps improve our efficiencies, reduces the cost as we create edits and test them. So most of you, I assume on this call are familiar with this, but if you're not, I just want to kind of do some level setting here. So I want to compare conventional breeding with what we can do with CRISPR-Cas or gene editing. So Alan touched on this before, is if we look on the left side over here, we have a disease resistance plant, but of what we'd call low quality. Usually that means it's not adapted for the area where we want to use it. And contrast this with something that's a high quality plant that is disease susceptible or sensitive. So by using traditional back crossing approaches, you can get to this state down here after 6 generations. That would be per disease gene. That gives you a high quality plant with improved disease resistance. So this takes time, this takes resources, this takes money. And what you end up with something that still carries 1% to 2% of low quality plant genetics or linkage drag, part of the genome. And this doesn't make our breeders happy, and it doesn't make our customers happy either. And so even though we have this process, you still have issues with linkage drag. So if we compare that or contrast that with a gene edited approach, again, we have the low quality disease resistance plant, high quality disease susceptible plant here. If we have that gene identified, we can basically in one step directly work with that elite germplasm or high quality plant and create some sort of product there that essentially has no unintended donor genetics to it. And I guess I can tell you one story here is that 3 or 4 years ago, I was out in the field with a plant breeder, and this plant breeder had developed a specific inbred. This was in maize. And he at the time looked at the inbred that he had created, and it was right by the particular genome edited that we had created. And he was just amazed at all the things that were good about the inbred he created, he could see in the edited, but also some of the warts too. I mean, there's no perfect inbred, there's no perfect hybrid. He could see that in the gene edited plant also. And so that really struck me at the time of just the power of gene editing, the cleanliness of gene editing and just what the possibilities were there. So what is the process of creating a disease superlocus? I'll keep this at a high level to go through kind of the steps here to familiarize with what we're doing with the DSL. So the first one here is discovery of native disease resistance genes. This is probably the most important step out of all of them. Since we are a multinational company, we have plant breeders located throughout the world. And we are in constant communication with them to identify sources of resistance germplasm that we can try to identify genes from that we could put into a disease superlocus. So we want to discover large effect genes that are not found in our elite germplasm. And so our initial foray into the disease superlocus is in North America. And so a lot of the disease resistance that we see is in tropical germplasm and comes from our breeders in the tropical areas. So we're not only looking for large effect genes, but we're also looking for dominant genes. And so this is important, particularly when we are creating a DSL in corn. So that's the first step there. Second one is gene characterization and evaluation in elite material. As I mentioned before, we do our discovery right in elite material there. We need genes that predict against different races that are out there. Again, this is targeted towards North America. And what our concept is using more than one gene per disease, and that is from a durability standpoint. And so for each of the diseases that I'm going to talk about, we have at least 2 genes that we're putting in a disease superlocus. Based on our modeling, we feel that's really going to improve the durability of the disease superlocus. Third one here is assembly of the disease-resistant genes into a DSL using gene editing. And so we've really pushed the organization for creating these large molecular stack constructs that we can put into corn, and just to make sure here that we're inserting, let you know that we're only inserting native gene sequences. So this would be a cisgenic product. So we use the same promoter. We're not changing terminators. When we put these genes in this stack, they are butted up against each other. So there's no superfluous DNA that's put in here. So taking essentially corn genes from tropical germplasm, identifying them and putting into elite North American germplasm, all in a single location. And then finally, the biggest thing is testing the efficacy to make sure that we don't lose any efficacy as we move it through this process. And then obviously yield testing of the disease superlocus in the elite material to make sure that we're not getting any yield drag. Okay, so that's a little bit background of gene editing from a disease superlocus process. Now what I'd like to do is focus in on creating a DSL in maize. And again, this is for North America, primarily the Corn Belt. Okay, so the overall concept here is have maize seed delivered resistance for our top diseases of interest. And so the idea here is to reduce the costs associated with fungicide application, kind of simplify the operation here. So customers will use many different ways to try to combat diseases. So use rotation of crops, will alter tillage, select resistant hybrids. Some of these are the different things that they've gone to. And so with the DSL, we hopefully we can simplify this process throughout the system. As I mentioned before, we want to achieve better yielding outcomes from this standpoint and reduce the amount of spraying that has to be done. So if you look in this wheel below here, this pie chart shows the estimated corn yield losses due to disease, and this is in North America in millions of bushels. So this has been split out into the different diseases here. So you have the foliar diseases kind of on the right. You have different rots, stalk rots, ear rots, other leaf and above ground pathogens. And so we looked at this list, and what we decided to do is focus on 4 of the diseases. So what are those 4 diseases? So if we look on the left here, we're focused on northern corn leaf blight, southern rust, gray leaf spot, and anthracnose stalk rot. And so we kind of picked these for 2 reasons. Number one, they are at the top of the list of diseases that have the most impact on yield reduction, and showing up there in that 318 million bushels due to these diseases. And second of all, too, is we also looked at it from a doability standpoint. So genes that were close to showing efficacy, that were dominant, that we knew could control these 4 different diseases. So if you look on the right part of this slide over here, stacking multiple genes in one location. So this cartoon here shows many genes, which are represented here by the different colors. There are many locations in the maize genome, and we have imprecise gene information on them. So the goal here to the right is having many genes that are in a single location here on a particular chromosome, and we have precise gene information on them. And so this is great for many different reasons. Some of them are shown down here. So this new breeding technique unlocks additional power of native genetics. So what does this mean? At the end of a season, when our breeders, he or she looks at the data that are collected for the year, they look at yield primarily, but then there's also these other secondary traits, one of them which is disease. And so if we can stack these genes together, put them in a corn against these diseases, that really either eliminates or essentially reduces how much selection pressure a breeder has to put on looking for these particular diseases. So in essence, what that does is that frees up the maize genome for the breeders to focus on other traits and yield. Another advantage of this is multiple disease targets. And so 4 of them are shown right here, and putting them all together in one locus, we think has a lot of advantage with it. So multiple native genes for each disease target improves resistance and adds durability. I touched on this before, is that we're not just putting in one gene, DR gene per disease, we're putting at least 2 in for each of these diseases. And then simplified genetics through gene editing accelerates plant breeding. So again, from a trait integration standpoint, this really improves the trait integration process. And so as we see this DSL overall, it has a lot of great advantages for the organization because the plant breeders are excited about it and the potential that it has. And we also think it has a lot of benefits for our customers. Okay, so got to show some pictures from the field. So on the left here is northern leaf blight, on the right is southern rust. And so in each of those pictures, on the left is the null, and on the right is a transgenic event. So the reason I show this is part of our testing here, is once we identify genes, we use transgenics to put these back into the plants to test their efficacy and make sure they're dominant. So here's 2 great examples here of NLB and southern rust, where we've identified efficacious genes that are dominant. Once we go through this point here, then we know that we're ready to use the editing piece. So to go into a little bit more detail here about the DSL assembly. So as I mentioned before, we're focused on one chromosome, putting these native genes into this one chromosome in elite germplasm. For NLB, southern rust, GLS, and ant rot, trying to find 2 dominant genes that we're going to put in there that will provide us efficacy and durability. So we're making DNA constructs here of anywhere to 3 to 5 genes, depends on the size of them. Again, only using native maize DNA sequence. So same promoters, same terminators, and putting button up against each other at this location in the chromosome. We're using SDN3 edits to insert into this disease superlocus. And then these edits or variants will be stacks, and we're using either sequential retransformation or breeding crosses. So the breeding crosses, I think, is pretty self-evident. Retransformation, what that means is that we go through a sequential process where we can only put a certain size of these genes into this location. Once we know they're in there, once we've done the genomics on it, once we've tested the efficacy, then we can retransform into that same background in order to put in the other genes from that for that DSL. So below, I just list a couple of technical challenges here. So from a vector construction standpoint and transformability, creating large constructs can be an issue, but there's a lot of things that we've forced the organization to try and tweak, and they've been great in responding in that we can get large constructs into the maize genome. And then the second thing is a retransformation process and what that offers and some of the things that we're learning from there. But in both of these cases, so far, we've overcome these technical challenges. Okay, so we created DSL variants. The answer is yes. So you can see 6 plants here. The 2 on the left are rust susceptible, so southern rust. The 2 on the right are northern leaf blight susceptible. And then the 2 plants in the middle are genome-edited variants that contain the genes for NLB and rust resistance. And so all these plants have been infected with either southern rust or NLB pathogens. We can see in the middle there is plants that are very efficacious against these 2 diseases. So we were really happy when we saw these plants here. We've moved these out to the field. We're testing them under field conditions. It recapitulates what we saw here in the greenhouse. We've also tested the hybrids under in various locations throughout the I-80 corridor, the Corn Belt. And what we can see is so far that there's no indication of any yield drag. So one other point I want to talk about is the advantage of the DSL compared to trait integration. So with the DR genes, we can use unlimited, that probably should be in quotes, amount of DR genes. But with trait integration, we know that we're limited to about 3 to 4 loci here. And so it really opens up the possibility here of things that we can do with disease, but also other traits too, as we work them through our genome edited process. As I mentioned several times already, reduces linkage drag. We do not have a variant created yet where we have all 8 genes from all the 4 diseases into one background, but so far all the yield tests that we've done with the genes that we have, we see no reduction in yield or linkage drag. We can deploy multiple alleles of the same gene, or we can deploy multiple genes within the same gene cluster. And so this gives us some advantages too as we go forward. We can adjust the DSL locus, and this is what's neat about the modularity of this. So swapping, adding gene alleles, and this is in case or when pathogen communities in the field shift over time. And so we're already working on what a next generation North America corn DSL would be, and so that we can take advantage of this modularity when this type of shift in pathogens occurs. And then the other thing is we can insert the disease superlocus in proximity to other traits or regions of interest. Again, coming back to this idea of trait integration, the closer they are together, the less number of progeny you have to look at to get something that is efficacious, doesn't create yield drag, and fits into the overall transgenic system that we have for insect traits and or herbicides, and with this genome editing approach. So in March of this year, we had a press release announcing that this process was coming. course, the question that always comes up is, when is this going to be commercialized? So we're in the process of right now of getting this 8-gene stack put together. We got to do a lot of testing on it to make sure it makes sense from an efficacy standpoint and from a yield standpoint. There's the regulatory piece to this that has to be there, but our hope is by the end of this decade, beginning of next decade, that we would have something that we could launch. So then another obvious question here is, what about a disease super locus for other crops? And so here's a pie chart here that shows average yield losses due to disease for soybeans, and this is in the US here. So again, a list of other diseases here and what their impact is on yield. And so we are starting a disease super locus in soybean right now that's targeted towards North America. So this is exciting. Hopefully this will go quicker because we have a lot of learnings from the North America corn DSL that we can apply here. So this is an exciting new opportunity for the disease group. So to summarize here, the disease super locus combines and repositions cisgenic disease-resistant genes to simplify farmers' disease management while protecting yield. And so we're really excited about this new approach here and what we think it offers to our customers and to our plant breeders. We have resistance genes that we've validated for 4 leading maize diseases that I've outlined here before. As I've just mentioned, the DSL facilitates trait integration and eliminates linkage drag. And we do have yield data from the field that shows this. But as I mentioned, we haven't put all 8 genes together and see what the impact is there. 4th bullet point, stacking resistance genes allows for protection against multiple pathogen races to provide a more durable disease resistance. And then finally, we're excited about this because we feel it's going to be an industry first product offering that, again, will have a lot of impact for our customers, really change kind of what it takes to control diseases, at least in North America corn environments. So Alan, I'll stop there, and I guess we'll do questions at the end.
Yeah, thank you very much, Jeff. This is super exciting, and again, I think complements the talks we've already had today. I mean, this is a great. We always talked about what people call SDN3, or really insertion or stacking of things, and thanks so much for for bringing this about and giving us an update of where we are. Yeah. So again, let's hold questions for Jeff. You can put them in the chat, write them down for yourself, to ask live, at the end of all the talks, and we'll ask, we'll ask. Sorry, we'll try to answer them at the end. So our next talk is Dr. Nathan Springer, and Nathan is head of genetic and genomic innovation at Bayer Crop Sciences, and he's going to talk to us about some dwarf corn that they've been working on. So Nathan, if you can go ahead and share your screen, I think
I'm working on it. Hopefully you can see that now. Are you seeing the full view?
Yep, perfect.
Excellent. Well, thank you for the opportunity, and thank you to all of you for being here to listen. You've heard a lot of fantastic talks already today. Mine's going to get even simpler than those. What I want to talk about is a short stature corn approach that could have been. When I was asked to talk about short corn at Bayer, honestly, at this point, we're not achieving short corn with editing, but I want to talk about why that is and what some of the power of editing in other applications is. So I'll start by a typical statement to tell you that I cannot predict the future. Any of you who know me know that quite well. And I want to remind everyone that a lot of, Alan gave a wonderful definition of the discipline of plant science, but a reminder that the purpose of plant breeding is really about how we solve challenges of producing a stable food supply for our population while protecting the resources of our planet. And I want to highlight the tremendous success that's been had in plant breeding. You know, if you look at productivity of corn systems and the amount of land required to produce equivalent amounts of yield, it's really shrunk over the years. And this is a testament to many different technologies and innovations that, together with plant breeding and genetics and agronomic practices, have really changed our production systems. So where does editing fit in, and how do we think about editing? Well, one of the ways we think about editing is how do we achieve a new trait? And you've heard about some pretty novel traits so far today. Changing the taste or flavor profile of leafy greens, changing peanut allergenicity, changing disease resistance. I'm going to talk about short corn, but I want to highlight that I really am just using short corn as an example. We're going to continue to shift some of the sets of traits and the production systems we use in agriculture. We've shifted over the years. One of the things that's not illustrated here, but that really changed, one of the successes in plant breeding was breeding for higher density tolerance. So we emphasized yield per unit area, but just by changing the system of how many plants and how close we planted them together. There haven't been a lot of huge management shifts over the last 30 to 40 years. The number of shifts that we're probably going to have over the coming 10 to 20 as we change, as the climate itself changes, and as we change policies to accommodate and develop more sustainable practices, we're going to be shifting our systems. So one example of a shift in a system is switching to short stature corn. So the Bayer product is called Preceon, and really, this product is based on simply shortening a corn plant. I will highlight that this is not a novel idea. This was the basis of the Green Revolution nearly 50 years ago. People have been thinking about using shorter stature corn or alleles that reduce height in corn since I was a graduate student over 25 years ago. The key is, how do we develop and go from that idea to a system that uses it? And I'll highlight a couple of the advantages of short corn. First of all, one of the key advantages is the idea of yield stability and the ability to tolerate substantial wind events. In these pictures here, you're seeing some images from field trials in 2020 when there were substantial windstorms in Iowa with wind speeds of 50 to 75 miles an hour. At the upper part of the picture here, what you're seeing is a region, a plot that was the short stature corn that had very minimal damage in this windstorm, while a traditional corn hybrid of full height exhibited severe lodging. So shortening the plant can allow for greater yield stability because the plants remain upright and you can realize the yield on the plant into the combine and into the grain bin. When you ask farmers about some of the major concerns they have, lodging events and substantial windstorms are one of the greatest concerns that they have. One of the other advantages that maybe wasn't immediately obvious about shortening the corn plant is the ability to have all-season access. So plants start shorter in the early weeks after planting, and they remain short throughout the growing season. This allows increased access to apply herbicides, fungicides, or fertilizer and a more on-demand setting. Rather than having to decide ahead of time, you think you might need this later, farmers can apply treatments on demand. So increasing yield stability, increasing access to the field are reasons that this system is a good idea. As I mentioned, this system has been proposed for many years. So why didn't it happen in corn earlier? Well, let's start talking about the ways we could achieve a system change. So we wanted to go from a whole population of corn plants that are relatively tall, 9 to 10 feet often, to plants that are less than 7 feet tall. We can use native traits. These are alleles or genetic variation within corn populations. We can use genome editing, and we can use transgenes. I'm really going to focus on comparing native traits, which all of the images you've seen so far were using a native trait or a naturally existing allele of a particular gene. There are solutions that can use transgenes, but I'm not going to focus on those today. I want to first talk about the native traits and how that works, and then why genome edits could be an alternative approach. So if we want to use deploy short corn, there are many different genes involved. One of the genes that was identified as relatively promising was a gene called BRCA2. So here in this picture on the left-hand side, you're seeing an image that you can see it at Maze GDB. This is the reference allele, one of the original alleles for BRCA2 that was found. So this allele and this mutation results in highly shortened inner nodes at the base of the plant, but relatively normal elongation at the top of the plant. This loss of function mutation does not change the reproductive architecture of the plant, so the tassel and the ear look relatively normal compared to wild type siblings. Now, I suspect most of you don't spend a lot of time in cornfields, but for those of you that do, you would say that this is not a particularly well-performing or strong agronomic corn line. This corn line is probably an inbred that was developed by plant breeders about 70 years ago. And many of the native alleles are in lines that the last time a plant breeder tried to improve them was 70 years ago, or in the case of some of those disease resistance alleles Jeff was mentioning, potentially hundreds or thousands of years ago as they come from land races. So one of the struggles is identifying the agronomic performance of these alleles that are in different backgrounds. One of the things I want to highlight about BRCA2 is that there are lots of native alleles existing. So this is just a partial list of alleles that have been characterized and are available through the Maze Genetic Stock Center. These alleles include some, shown in the picture in the middle, that is a single base change, so a single nucleotide change can result in a BRCA2 phenotype. There are also examples of alleles that are thousands of base pairs of insertions. These are transposons, and some of these have been identified as spontaneous insertions. So in a large population of maize plant, a single individual of short stature is identified, and this individual had a novel mutation that occurred without intentional mutagenesis. I will point out that many of these native alleles are due to transposon insertions, so large sequences that inserted within the BRCA2 gene. However, most of these mutations are functionally equivalent. That is, they all result in a loss of function of this gene that results in a shortening of the corn stature. So we have lots of BRCA2 alleles. Some of them have been characterized over 50 years ago. Why are we just turning this into a trait that makes it into farmers' fields now? Well, for one thing, you have to have a management system that utilizes it. So developing that management system takes some amount of time. But you also have to deploy it into improved genetic backgrounds. Jeff highlighted this very nicely, that it's not just determining the effect of an allele on a particular trait, but you have to move that allele into the right genetic background so it will perform in the right environment to test how that allele actually performs. So we want to deploy a BRCA2 native mutation. Now, this mutation starts in this genome, and I've used a kind of awful green color on purpose because it's highlighting that this genome is not a well-adapted or elite genome. And I want to move this allele into 4 different elite inbreds to test the performance and ask what will happen in terms of agronomic potential. What I'm hoping to recover is this. What I want to recover is a genome where the only change is the introduction of this novel allele. And I'll highlight, Jeff did a great job of this. This is going to take at least 2 or 3 years, multiple generations, and substantial resources to create these lines. I don't get what I want, though. What I get is something that looks a bit like this, where the BRCA2 gene was brought over, and this particular allele, but the region surrounding it actually is derived from that low-quality parent. And often there are 1 to 2 to 3 other regions of the genome that are derived from that parent as well. So some proportion of that genome is making it into my line and has the potential to affect performance. And this took several generations. Now, plant breeding is absolutely fantastic, but I want to highlight some of the limitations. So the first one is that crosses take time. Every time we put together alleles from or traits from different lines, we're making crosses and we have to go through a selection process. We're also trying to maintain favorable linkages. So as breeders select for a line, they're creating a set of alleles that operate together to maximize a large set of traits. Often, and I'm guilty of this today, I'm focusing on one trait, which is stature. But remember, these products are being tested in environments where they have to maintain a trait of interest together with resistance to pests, high yield, abiotic stress tolerance, flowering at the right time. All of these things are multigenic and require breeders to be selecting on the alleles to give them the performance they want. When I want to change even a single trait, like adding a brachytic 2 gene, and I make a cross, I'm often taking a card out of the stack, and at least part of the deck falls apart, and I have to rebuild it. Plant breeders are excellent at rebuilding the stack, but it does take time, it's resource intensive. One of the other key issues, and this is seen with brachytic 2, is that we get unexpected results. We often know the effect of a particular allele in a certain genetic background. So, and we might know another background. We expect if we add that trait to some other background that 1 + 1 will equal 2. However, we often get unexpected interactions of an allele with a genetic background such that adding the sums don't add up. We get less or more of the trait in some backgrounds than we might have expected. This is true for most native alleles and most processes that we would backcross an allele into a line. I want to highlight that while it might sound like I'm saying plant breeding doesn't work all that well, plant breeding is highly effective. For short stature corn, it took a while, but we have introgressed a native allele to produce hybrids that are high performing. So my comments are not to say that this approach is unfeasible. They're simply to say that there's another way to think about things. So as we think about editing and the potential for editing, you've heard some wonderful presentations today about creating novel traits. What I'm going to talk about, though, is not about creating a novel trait. I just told you there are 20 to 30 native alleles of brachytic 2. I don't actually need editing to create brachytic 2. So why am I still thinking about it? Well, all of us are sitting here right now in rooms spread across, I don't know what geography. I know at least California to Washington, and we're connected. We aren't generating any new information today. We're just sharing information, and that's really the power of the internet. The internet, by and large, as a function, is not generating new information. It is changing the pace at which we share or move information. For thousands of years, if a plant breeder wanted to move a trait from one line to another, it required crosses and a fairly time-consuming process to move that trait. Editing offers the potential for a faster movement of information across our germplasm or populations. And that pace of movement has real potential, just like we've seen with the internet and how it's impacted all of our lives. So just a reminder that what we're talking about here is really the simplest type of editing. Alan gave a great overview of the many different technologies and approaches you can use. You just heard a very clever application from Jeff about how we can use genome editing machinery to target insertions and move alleles to a particular location of the genome. What we're talking about here is really the very simple process of targeting a Cas enzyme or a CPF1 enzyme using a guide RNA to a precise genomic location. At this precise location, you cut the DNA, and you rely on the fact that the repair of DNA by the cellular machinery is not always precise. And when you get imprecise repair that doesn't exactly fix the error, part of that target site is removed, often creating small insertions or small deletions. So we can edit the brachytic 2 gene. This was an experiment done by some colleagues several years ago at Bayer. So in this case, we started with a relatively elite inbred, and we used a single guide RNA, and we caused a single 1 base pair insertion at the target site. This results in a loss of function because only a partial protein is produced. We compared this to a native allele. The native allele was a 4,000 base pair transposon insertion that had occurred spontaneously. This transposon insertion had been backcrossed in, so it went through that backcross or integration process. Functionally, these two alleles result in identical plant heights, even though they're quite different at the molecular level, because they're both a functional knockout. The difference is the transposon allele that was backcrossed came with some information from the genome in which it arose. But essentially, we're creating equivalent traits through editing. Now, not only can we edit in one line, it's quite feasible to create edits using the same machinery, but introduced into multiple different genetic backgrounds. So in this case, this was an experiment. This was actually not targeting the BRCA2 gene. This was editing a different single locus. But there was a simultaneous transformation of 22 elite female corn germplasm lines that represented diverse genetic backgrounds. These are, in this plot, I'm simply showing you lines from different relative maturity groups, from relatively early flowering to relatively late flowering, and the ability to create edited events across each of these different types or germplasm sources of corn. So we could go in and independently introduce edits at the same locus. So we essentially take these, in this case, we'll go back to our illustration of 4 elite lines. We introduce the editing machinery concurrently in each of these germplasms. And the identical, one outcome could be achieving identical edits in each background. So after essentially 2 generations, one generation where the transgene is present and able to do the editing, and a second generation where you remove the transgene and homozygose the edit that was created, you could achieve an outcome where after lots of screening, you would identify an identical outcome, maybe a 5 base pair deletion that's present in each of these lines. Alternatively, you could end up with functionally equivalent outcomes. This is much more likely to happen where you targeted that guide RNA to exactly the same site in each of the genomes, but the repair process in some cases gave you a 5 base pair deletion. In another line, maybe it gave you a 13 base pair deletion. Functionally, these all result in a loss of function for the same gene, but the molecular basis for each of these alleles is distinct. Now, the advantage to this approach is instead of taking 6 or so generations and bringing additional genetic information with, in this case, it requires 2 generations and your edits are rapidly deployed into the background of interest. So this gets back at that concept of the information sharing or the movement of information across genomes can be enabled and occur in a much more rapid fashion through genome editing than it can through traditional crosses. So I'll highlight that there was a recent publication that thought about this, not necessarily from the context of corn and short corn, but in this case, it was an example of citrus greening. And really was highlighting the difference between the current state of how we think about regulation of genome edits and really focusing on a specific edit or a specific event, the way we think about transgenics, where that is created once and then moved across germplasm through crossing. That would be a way to use editing to create novel traits and then cross them. Alternatively, as we start thinking about directly editing into diverse germplasm, we think about a collection of germplasm in which we want to shift the trait in all of that germplasm from one state, in this case, susceptibility to a disease, to resistance to the disease. We would go into each germplasm, independently create edits that could either be identical or could be functionally equivalent. So different molecular changes, but at the same locus of the genome. Thinking about how we regulate and how we handle these collections, a collection of functionally equivalent alleles rather than a specific event, will be important to think about how we enable genome editing to potentially move alleles across our population, our germplasm, in a much more rapid way than we do currently. And with that, I just want to reemphasize that the reason we want to do this and we care about that pace and speed is that there are critical problems we need to solve with our crop products. These problems are the same whether you're at any of the institutions or companies that are talking with you today. And part of the challenge is how do we use these new technologies and the opportunities they afford to be more nimble and agile in how we approach creating our products. And so with that, I will say thank you, and I will come back in a little while to maybe answer some questions or maybe not give answers, but just make the situation a little muddier.
Thanks, Nathan, again, to nice complimentary talk on use potential of gene edit and how that might change plant breeding. So please stay around for the questions for the speakers. Our next speaker is going to be from Dr. Rachel Egger. She's a principal scientist in reproductive biology in Science Leaf for new breeding and seed production technologies at Syngenta. And she's going to talk to us about really gene editing, not necessarily for a product, but also, how can we use it in breeding per se? Rachel, go ahead, share your screen.
Okay, let me get it pulled up. You guys should be seeing full-size PowerPoint now.
Yep, perfect. Thanks.
Okay. All right, I'm very excited that Nathan and Jeff gave us such great introductions to these topics today, because we're going to touch on a lot of the same themes that we've been seeing earlier in the session. So it's really been a great session so far and teased this conversation on nicely. The topic I was asked to present on today was haploid induction and how we're using it to accelerate breeding here at Syngenta. And specifically, I'm going to touch on how are we combining haploid induction with genome editing. But before I get too deep into that process, I wanted to really start at the beginning. Let me just hide the meeting control. There we go. With thinking about what is conventional plant breeding and how far have we come. Now, I won't spend a ton of time here. Obviously, you guys have already talked about, where plant breeding fits into the scheme earlier in your sessions today. But fundamentally, the science of changing plants to have desired traits through selection on variation is where plant breeding began. And I like this example because it's both delicious and diverse, which is that Brassica oleracea is a plant that's native to the Mediterranean. This is what it looks like. It's kind of a little weed that grows on rocky coastlines. But over thousands of years, starting with the Romans, actually back in the 6th century BC, humans have actually selected positive traits that we like in brassicas to give you the diversity of cabbages and broccolis that we have today. And the reason that that's possible is because there's this underlying variation in the genomes in the original populations of the brassica that we can actually pick and choose amongst the differences in that variation to end up with very different outcomes. Obviously, a Chinese cabbage and Brussels sprouts look very little alike today. So if we follow that concept forward, I love the statistic that came out of a publication in 2019, which is that if you're looking at a hectare of wheat, maybe you're in North Dakota, looking out across one of those fields, any given hectare will have 20 billion new mutations to the genome. So mutation is something that's happening in the genomes all the time, resulting in that variation that we can then use for plant breeding purposes for selection for new traits. And if you imagine looking at this particular field, that means that if you were to select any one plant, there is a decent chance that you'd pick one that has a change to its genome, a mutation represented by one of these red stars. In some cases, what you would see then is that those mutations actually lead to new traits, changes in phenotype that we can see in those plants. This is an example out of a study in 2018. In this case, they actually increased that rate of mutation using X-ray irradiation. There's also chemical mutagenesis techniques. Both of those are non-GM technologies that have been used for a long time in plant breeding circles. And when you have mutations that occur that result in changes in phenotype, what you'll see is that you might start to get differences in grain, size, and color, for example, or changes to the morphology of the spike, fewer bristles, longer spikes. Depending on what outcome you're after, you would select amongst these phenotypes for the traits that you want to move forward. But this approach it's very non-specific. So you're making lots of changes to the genome in a genome-wide fashion, and then you screen based on phenotype to pick what you want to move forward. And we're now in an age where we have the access to tools that can do this in a more precise fashion. So fundamentally, when we think about what changes we're making with genome editing, most mutations that you find in the genome, either with gene editing or another technology, are going to be small. So if you're doing this in a non-specific way, you're going to see a lot of what we call indels, which are insertion deletions and substitutions. If those fall in a non-genic region, you're not going to see any change happen to that plant. But when you're lucky enough for those to happen in a gene, and with genome editing, we can target these to a gene, that's where you're going to actually result in changes like amino acid substitutions, protein loss of function, and gain of function, where we'll really see those changes in traits that we're after. There's also the possibility to target regulatory regions where you can change gene expression. A good example of this that we see in plant varieties pretty frequently is in the color pathways, so anthocyanin expression. Often, if you're looking at elite commercial corn, you're not going to see a lot of purple leaf tissue, right? There's not purple fields of corn out there in the corn belt. But just by changing expression of those genes so that you're able to make those pigments in the leaves, you can get purple corn pretty easily. And that's true in a lot of different species. You'll see it in your ornamentals as well, of course. In rare scenarios, your mutations are actually going to be larger structural variation changes. So you might have something like deletion of a large section of a chromosome, a duplication event, maybe even an inversion, or translocations and insertions where you have large sections of chromosome that'll actually move from one chromosome to another within the genome. These are often events that can be the start of separating two varieties within a plant species or even subspeciation. And the example on the right on this slide actually comes from two subspecies of rice grown in Asia, where you've got each of these vertical columns is a chromosome that corresponds to the one next to it. And you can see these areas I've highlighted in pink kind of form an X. That actually is places where there's a real inversion between these chromosomes in these two subspecies, and it has contributed to their subspeciation. Because when you have these larger rearrangements, you're essentially making a lot of changes all at once, which means that you're ending up with plants that are going to have more differences between their traits, greater variation between them. Okay, so moving forward in time, thinking about how do we do this in a more modern way, the way that we we kind of stack the deck as plant breeders, right, is that we would take two parents with different traits that we actually are interested in combining and cross those two parents together to make our F1 hybrid. So in this example, you can see we're using corn as an example. Parent A has longer leaves but fewer roots. Parent B is the opposite. We cross them together in the F1 and we see that the parent B phenotype is the more dominant phenotype in this hybrid. And then if we go another generation and self this plant, we'll have recombination between the pink and the blue chromosomes happen between these generations, resulting in segregation in this F2 generation. So now you've got plants that have characteristics that came from each parent A and parent B. And we could actually go through this population and pick the outcome that we want. So if we want long leaves and lots of roots, that's going to be this plant in the middle. You could also do this genotypically by selecting on the DNA if you know which region of the chromosome is associated with this trait. So for example, if we know that having the tip of this chromosome from parent B, the pink parent, would result in better disease resistance, for example, we could use molecular DNA markers to make that selection without having to actually look at the traits directly in the field. So that can greatly accelerate things and reduce the amount of screening you have to do as well. Now, the next natural step of this, and Jeff outlined this beautifully earlier, would then to be identified in your segregating population that you want to get back to inbreds for hybrid production if we're doing corn, or true breeding lines if we're in another crop where we're going to do single seed descent instead. You're going to have to do a series of either selfs or back crosses to get back to that nice homozygous allele state. So you can see here in the hybrid, we've got different banding patterns on these two chromosomes between blue and pink. And then as you make this series of crosses, you're eventually going to get back to where you have pretty close to exactly the same on both chromosomes. It's not going to be perfect or something in the 97 to 98% identical. This takes time, this takes money. In corn, we're talking about a couple of years of generation time. And so haploid induction has really come about as a shortcut to do this faster. So you're going to have just 2 generations of time to get to that homozygous outcome, as opposed to the 6 or 7 generations that are diagrammed on the top of this slide. Now, there's a few different methods you can use to get haploid induction. In corn, you're typically going to see a haploid inducer line used as the male parent in a crop. And what that is based on is actually almost all haploid inducers in corn come back to this line, stock 6, which was first identified by corn breeders in the United States in the 1950s. It was then used by breeders for decades without fully understanding why in haploids. And it wasn't until 2017 that a group at Syngenta, as well as some academic groups around the same time, actually identified the gene responsible for haploid induction in corn. Very briefly, the way this is going to work is that the haploid inducer in this cross is going to be your male parent. You cross it by a female that has the genome that you're interested in. So that would be something from that segregating population we saw on the previous slide. And then some proportion of the progeny of that cross, roughly 5 to 15% for most haploid inducers, is going to have only the genome of the female parent. That male haploid inducer genome has been lost entirely. The trick here, though, is that we actually have to get this back to being what we call a doubled haploid or a diploid, because haploids are intrinsically infertile. They're not able to complete meiosis successfully. They will never make pollen. So we use a mitotic inhibitor to actually get this to double its genome back to two copies of the genome, a nice functional fertile diploid plant that we can then use as a breeding start in the next generation. So I want to walk you through a little bit of the work that was done to actually clone this gene back in 2017, because it's also a great example of how we can use genome editing as a research tool. So the genetic mapping of the haploid trigger was done by a team of folks at Syngenta in 2017. And there were a couple of breeders that had inducer lines to do association mapping to ask which regions of the genome could we connect and correlate with this haploid induction phenotype, essentially, this haploid induction trait. And they had actually gotten down to a pretty narrow interval on chromosome 1 that contained only 7 gene candidates. And so when we went through and sequenced the DNA in each of these genes, what we found is that there were these two highlighted in red, that had obvious mutations that were different between what we would call normal corns. This is anything that's not a haploid inducer, and in our haploid inducer lines. The first of these genes, MATL, had a 4 base pair insertion in the 4th exon, resulting in a premature stop of the protein. So you can see that this is the protein sequence on the bottom of the slide, and this one here actually, after that insertion, ends up with a stop codon much earlier than it should. So it makes a much shorter version of the protein. The PGM gene was actually missing entirely in the haploid inducer lines, which obviously could potentially cause issues as well. We didn't know which of these was functional and causative for haploid induction because we didn't understand how haploid induction worked at the time. So the experiment was set up where we had a transformable haploid inducer that one of our breeders had created a couple of years prior. This was a stroke of luck and foresight on his part. It was not something at the time that we knew that we needed, but it was available to us because he had thought that it might be useful someday and made it as part of a breeding program. And so what we were able to do was to take that transformable haploid inducer and ask which change in the DNA was actually responsible for what's called here the DNA disappearing act or haploid induction. So this is often what a haploid induction ear will look like, because in addition to getting haploids induced, you will also have some embryo abortion that happens as sort of a side effect of the process. So we often look for this spotty phenotype to know that our haploid induction is working well. We then took the transformable haploid inducer and added back a wild type copy of the PGM gene, which remember was entirely absent in our haploid inducer lines. And what we saw was that the ears on those plants still looked like haploid inducer ears. So adding back the wild type copy of this gene did not seem to complement our phenotype. We then tried the same experiment, adding back a wild type copy of the matrilineal gene. And what we found is that that resulted in a nice normal seed set on our ear. And in fact, we did the reverse experiment using genome editing, where we took a normal corn plant, something not a haploid inducer, targeted a guide RNA and a Cas enzyme to make a break in this MATL gene, and saw that that plant then became a haploid inducer, where we saw this spotty kernel phenotype and confirmed that we had haploids amongst the progeny. This is very exciting. We then wanted to know, we finally know what the haploid trigger is. What gene is that? What does it do in the plant? So matrilineal, as it was named, is now, we now know it's a sperm cell localized membrane phospholipase. So it's involved with creating the membrane that surrounds the sperm cells, and then their eventual fertilization and fusion with the female egg cell from the other parent. And we actually were able to add a GFP tag to the matrilineal protein and confirm where it was expressed in the plant. So what you're looking at here is a section through a pollen grain on a confocal microscope. And the green that you're seeing, these kind of two crescent shapes in the middle, those are the sperm cells inside of a mature pollen grain. So you can see they're nice and bright green, lots of matrilineal protein expressing in that membrane packet. You can actually follow those sperm cells as they move down the silk towards the female side, and then see that expression is actually directly transferred to the fertilized embryo after fertilization and fusion of the sperm and egg cells. So this made a lot of sense for haploid induction. It's directly related to what happens during those early embryo stages and delivery of the sperm cell. Now that we knew what gene caused this in corn, we were able to take that concept and directly transfer it to other crops in the grass family where matrilineal is closely related to the copy that we see in corn. So this is an example that was published in rice, where we were able to say, you know, MATL based on synteny and how it lines up to the rice genome is probably one of a handful of phospholipase type A genes. We can see a lot of conservation in the protein sequence. That's this alignment that you see over here, where we have nice chunks of letters that align well between those proteins. And when we looked at the expression pattern of where it was expressed in the plant, because
I just want to touch base on doing on the seed.
So what we saw then was that this particular PLA1 enzyme, which is the ortholog to MATL in corn, was well expressed in a particular stage of the panicle, the flower of the rice plant, which made a lot of sense for packeting up that sperm membrane again and to align developmentally with the right timing. By editing this gene, we showed that we could then make a haploid inducer in rice, where we got diploids and then, as expected, the smaller haploid phenotype out of the progeny of that cross. And then this work was actually published in 2019 to show that you could do this, at least within the grasses, to edit matrilineal and make haploid inducers in other crops. Coming back to our overview diagram then, so we now know more about how haploid induction works in corn and in other species, at least within the grass family. And then we've been actively working to improve this process in production by focusing on a number of different targets of the process. Part of those is understanding what other genes are involved in corn and other species, other ways to derive haploid inducers, thinking about running breeding programs to increase our haploid induction rates. So matrilineal alone only gives you about a 5% haploid induction rate. We like it to be much higher than that and have more successful lines than that now. And working on improving this doubling step, because this is not a perfect process. And so there are efficiencies that can be gained here as well. But the key thing that I want to focus on today that we've also added to this process is asking, could we combine this haploid induction cross with genome editing? Now, some of you are wondering, why would we combine this with genome editing? Maybe that doesn't immediately seem to have value. And this idea came about because Syngenta is and was actively developing genome edited crops in a number of different species. You've heard lots of talks today about how that can bring new value and new traits to the market. But the key distinction here is that while we have the technology for genome editing, many elite commercial crop varieties are not transformable. And that is especially true as you move to some of the crops that are not as frequently transformed, right? Where you might have one or two varieties that you can transform well. In corn, we have a little bit more flexibility, but there's still varieties that are going to be recalcitrant. And if you don't have the ability to transform something directly, and Nathan talked about this in his section of his talk as well with the citrus example, what you're going to end up doing is editing something that is transformable and then taking those edits and introgressing them through that same 6 or 7 generations of back crossing into your elite lines that are non-transformable. This takes too long, it's too expensive, and it sort of removes a lot of the advantage of doing genome editing. You're getting a more precise product, but you could possibly reintroduce that linkage drag by having to do this with introgression if you're in crops or varieties where you don't have the ability to transform all your elite varieties. So we wanted a method of editing commercial varieties directly without having to go through transformation. You've all heard lots about CRISPR today. I won't spend too much time here, but fundamentally, there's a couple of pieces of machinery you need anytime you're going to make a CRISPR edit. You need a Cas protein. That could be a number of different options. It needs to pick up a guide RNA that we will use to target the right section of the genome. And then after it makes a cut, that imprecise repair machinery comes in where you're going to get a diverse outcome of indels. And those are actually what we're interested in this case, because we want to have a different outcome than what was there before. So our process that we have here at Syngenta, we now call high edit, where high stands for haploid induction combined with editing. And the fundamental backbone of this is that you need to have a transformable haploid inducer to enable this process and make it possible. So you start by transforming that haploid inducer, putting those CRISPR components into it. You're then going to pollinate the lines that you want to actually have both haploid induction and editing happen in simultaneously. This line does not need to be transformable. It can be any elite that you can cross by this haploid inducer. After that, you're going to sort out the haploids, grow them up, and actually check their DNA to make sure that the editing happens and select which editing outcomes you want, because you'll have a small diversity to select amongst. And then you're going to self those plants and put them into the breeding pipeline to actually check how well the traits are working and to benchmark them against other things that are in the breeding pipeline. If you want to look at how this works at the cellular level, and this is really my bread and butter as a reproductive biologist, that male haploid inducer, because it's transformable, it's going to carry the editing transgene. So that's the Cas enzyme and the guide RNAs that we're providing to the plant. That comes in riding in the sperm cell because it's been transformed into its genome directly, which means that the sperm nucleus already is able to express the Cas enzyme and the guide RNA. There is no CRISPR machinery coming from the female in this example because the female is our elite line. It could be non-transformable or transformable. It doesn't matter. We're not going to transform it in any scenario here. And once the sperm cell comes down that silk, as we saw in that GFP image, and fuses with the egg cell, what you actually have is a hybrid zygote, meaning that both the male and female genome are present. The male genome, because it has that transgene, is able to express Cas12 and the guide RNA, but that guide RNA and the associated Cas enzyme are free to target either genome, the male or the female. They're present in the same nucleus. And so as long as that guide RNA matches a sequence on this female chromosome, it's possible for Cas12 and the guide RNA to actually find that location and make this edit represented by a red star. This is also the same stage where haploid induction takes place. So this male genome, the chromosome containing the transgene, will be eliminated during haploid induction, resulting in a cell that only contains the female genome. And once we take that through the doubling step, what you will actually have is a double haploid that has homozygous edits, only the female genome, is transgene free, and was never directly transformed. So this is pretty exciting technology, and there's lots of things we could do with this. And we developed it in corn because we had the resources available to us and a robust double haploid process in corn. But we'd like to extend it to other crops and be able to do this in lots of different applications. So that requires having haploid inducers beyond just the matrilineal gene, other types of haploid inducers that we can have in other species. That's where SENH3 comes in. This is a gene that's part of the kinetochore complex, and it's well conserved across all plants and actually into animals as well. So it's a highly conserved protein overall. And there's been quite a bit of work to show that you can derive haploid inducers from mutations in the SENH3 gene, although it's not trivial to do in any one species because different mutations seem to be required depending on the particular allele of SENH3 that a species carries. There's also an additional bonus here. If you do a SENH3 haploid induction cross, you can use the parent as either the male or the female in the induction cross, which means that you have the opportunity to do cytoplasm conversion if you use it as a female inducer, because it'll keep the cytoplasm from the egg cell that came from the female parent. So we, in 2016 and then extending into 2018 and 2019, did some experiments to make a SENH3-based haploid inducer in wheat, where we showed that we could do what's called a tail swap experiment, where you change one part of the proteins in SENH3 to make it sort of semi-non-functional. And we were able to show that when we used that tail swap mutant to make haploid induction crosses in wheat, we did get these nice little plants that if you run their genomes through a ploidy analyzer and compare genome content, that's these charts on the bottom, what you'll see is that a normal diploid should run out with 2 peaks at roughly 100 and 200, but a haploid shows up with a peak at 50 and 100. So half the genome content, and that was in fact true haploids that we saw in these small plants out of these induction crosses. So we were able to make a SENH3 wheat haploid inducer. There are some species where folks have tried to make SENH3 inducers and have not been successful. One of those for a long time was maize. But there was this report out of Kelly Dawes' lab at UGA in 2021 that they actually figured out a simpler solution to this problem. So it was thought that some of these species could not tolerate a null mutation in the homozygous state for SENH3. So you needed a functional copy of SENH3 to be alive and be a plant, essentially. But what Kelly Dawes' lab showed was that if you had a null mutant knockout in SENH3, but you maintained it as a heterozygote, that was enough SENH3 to do normal plant operation, but also enough of this mutant copy that you could get a small amount of haploid induction, so roughly a 5% haploid induction rate. So this was a pretty exciting result to show that you could expand this into other species where previous SENH3 efforts had been unsuccessful. So we're currently working on expanding SENH3 into other species beyond that as well, that will have multiple haploid inducer options available for the high edit process. And this will be the last slide that I want to end on today, which is that we've heard some exciting examples of using what's called SDN3 editing, so editing that DSL stack earlier that Jeff talked about. And there are indeed an expanding number of things that you can do with genome editing. So the example I showed you today was just doing a simple cut in a gene that typically will inactivate a gene if you do it in the coding sequence and make those indels. But there are lots of other things we can imagine doing in the future. Modifying a gene by just changing a few base pairs to change a single amino acid in a protein, for example, changing gene expression or removing a gene entirely, as well as doing replacement or insertion are all things that we should be able to do with this hybrid system in the future. And I will take questions at the end when we get back to the live session.
Thank you very much, Rachel. I mean, I think very clear, at least to me. And also highlight, I have to say, highlighting a UC Davis technology, which is the SENH3, which was done by the late Simon Chan in 2010.
Absolutely.
Yeah. And okay, so our next speaker is my colleague, Dr. Imtiyaz Khanday. And Imtiyaz joined UC Davis as an assistant professor in 2021 as a professor in the Department of Plant Sciences, specializing in plant reproductive biology. And today he's going to talk to us about something that I think people have been, I know I took what Apomixis was when I was an undergrad student and sort of forgot about it because it's a difficult technology. And Imtiyaz, I think, has figured out a way to make it real. And Imtiyaz, go ahead. I think just switch your screen to full screen, and I think we're good. That's
okay.
Good.
All right. Okay. Thank you, Alan, for the introduction, and thank you for having me. The previous speakers made my life very easy, talking about why we need gene editing in crops and also how it works. So I had to make some last-minute adjustments to my presentation to reduce the redundancy. So I'm going to focus on Apomixis and tell you why we need it in crop plants. So Apomixis, it's a natural mode of reproduction in plants. It's an asexual mode through seeds. So you can have asexual reproduction through other meanings like cuttings and others, but this happens through seed. And a lot of plant species have figured out a way that, how they can make seeds asexually in the absence of meiosis and fertilization, which are the two fundamental processes involved in sexual reproduction. Okay. So a plant is essentially able to make clones of itself. We know about Apomixis for a long, long time, from 1829. That's at least the reported, the first reported instance of it, where a single plant from Australia, the native holly plant, was introduced into UK, and it was able to make seeds on its own. Now the sexes in this plant are separate; the male and female plants are separate, but it was still able to make asexual seeds. In nature, it can occur through different modes. It can be sporophytic, it can be gametophytic, depending upon where the apomictic initials come from, but I'm not going to get into the details of that. So it is a pretty widespread, as I said, mode of reproduction in many plant species, to be precise, more than 400 angiospermic species, But the point, the problem is it doesn't exist in major crop plants. It's known in some forage grasses and, say, for example, in some citrus species, but it doesn't happen. It's not found in crop plants. So, but why do we care about apomixis in crop plants? Is for this reason, which is, hybrid vigor preservation. So we know in many crops we have, when we cross certain inbred varieties, that results in the enhancement of certain quantitative characteristics, yield being one of them, or it can be biomass, but what's called the hybrid vigor. So I'm showing here just the yield, so because it looks good. But what happens is that these high yielding hybrids, undergo sexual reproduction, and then that results in recombination and segregation of these hybrid characteristics, and this hybrid vigor is actually lost in the next generation. So hybrids stay hybrids for just one generation. But if we could somehow make these high yielding hybrids to undergo apomixis, then we can produce clonal progenies and we can preserve this hybrid vigor over generations. Just to give you a brief account of why we need hybrids. In no other crop has hybrid vigor been exploited as much as in corn here. Yeah. This is the time, around 1920s when we started to make hybrids. And after that, you can see we have seen, at least here in the US, exponential growth in corn yields. And this is all thanks to these hybrid corn. About 95% of our corn grown here in the US is hybrid. And this is mainly because it's relatively easier to make these hybrids in corn. You have already well-defined inbred lines that you cross together. And then the morphology of plant also helps. You have separate male and female inflorescences. So you can easily detassel corn and then basically pollinate it with the other inbred variety and create the hybrid. But this is not the case in other crops. Say, for example, in wheat, rice, sorghum, and other cereals, the floral morphology is very complex. You have both male and female pods contained within the same flower. So emasculating and creating hybrids is a difficult process. So which usually right now involves a 3-parent cross where you have a male sterile line, you cross it with a maintainer, and then create a hybrid that you indeed cross with the restorer line to create an F1 hybrid. And this results in really huge costs for this hybrid seed production. And as I said, this is the case in rice, wheat, and others. Just to cite an example of what hybrid crops, what impact hybrid crops can have on crop production, I'm citing this example from India and China. So China uses only 30 million acres of land for growing rice, whereas India uses 1.5 times of that, about 45 million acres. Whereas rice production in China is almost double that of India, which is 210 million tons, and India produces only half of it. The reason for this is that, and it's not 100%, only 50% of the area in China is actually under hybrid rice cultivation. So hybrid crops can have really huge impact on crop production. Having apomixis in crop plants would not only enable the preservation of hybrid vigor, but it can also make our breeding programs faster. So you have any elite genotype that comes out of a breeding program, you can immediately fix it and you can basically use it for propagation. This has huge implications for breeding programs, whether they are done in a private commercial seed company or through other academic breeding programs, where currently, if you have a new hybrid, it takes about a decade, maybe more, to run it through the standard pipeline to ultimately be able to bring it to the market. But if you can fix it immediately, you can do it in a year or maybe 2. It will make our breeding programs cheaper as well. True hybrid and commercial products now we don't need those complex crosses anymore that I just showed you. We no longer need to maintain those parental inbred lines continuously, so we can save a bunch of land and labor. And it will also enable the propagation of currently vegetatively propagated true seed crops, say for example potatoes, which has other implications, which is it will enable a disease-free propagation of these vegetatively propagated crops. And same holds true for cassava and other vegetatively propagated crops. So for all of these reasons, apomixis in crop plants has been called as a holy grail of agriculture. It's a breeder's dream. So as I told you, we know about apomixis for a long, long time. We knew about its potential in preserving hybrid vigor as well. So the obvious choice seemed to be that we can just cross it into our crops. Now, as I said, it doesn't exist in crop plants, but it does exist in some of the relatives of these wild relatives of these crop plants. Say, for example, in millets, in wheat, in maize, though there are no known wild relatives in rice which are naturally apomictic. From these crosses, it didn't make much success, although we were able to bring this apomictic trait into these crops, but back crossing and gaining back those chromosomes, crop chromosomes, it usually disappeared. And now back then, I think one of the reasons for it not succeeding was that we didn't understand apomixis that well, but we have made tremendous progress in that area in the last few years. And I hope that at one point we would be able to integrate natural apomixis into crop plants. But for now, I'm going to tell you about synthesizing apomixis in crop plants. Now, we have no relation to working on apomixis whatsoever, but what we were investigating was the opposite process, which is sexual reproduction. So in sexual reproduction, we were studying what's the gene expression in gametes, that's egg cells and sperm cells in rice, and how that changes after fertilization in the zygote, and how that change in gene expression or the contributions of that gene expression, whether coming from male or female parents, affect the zygotic development. So I have skipped a lot of data, a lot of studies here, but what I'm telling you is the crux of that study, just one of the points, basically, not the whole crux of that study, is that we found this one gene called baby boom 1 or BBM1 that it expresses only from the male alleles after fertilization. So the genome that you basically inherited from sperm cells, this gene shows expression only from that genome, which is shown in this SNP analysis here. That were basically this SNP analysis was carried out in a hybrid genome of an indica and a japonica rice. But hybrid genomes have erratic gene expression, so we wanted to make sure that this was a real thing. So we also created a baby boom GFP line of which we used as a male or female parent. So what you see here is that when we use this GFP line as a male parent, we can see the expression in the zygotes, but when we see this, when we use this GFP line as a female parent, there is no expression. So confirming that baby boom after fertilization expresses only from the male alleles. Now, this was very interesting for us because this parent-specific gene expression is very common in the endosperm in seeds, but it's very, very rare in the embryonic tissues. The next thing that we looked at is, when does this expression start? Does it start before or after fertilization? So if you look at the transcripts of this gene, we can detect them in the sperm cells, but there is no expression in the egg cells. And this is shown here, that is not only transcribed, but also translated before fertilization. The two basically dots highlighting here are two sperm cells showing baby boom expression. So what we also did was, if we ectopically overexpress this gene in rice, what we see is that it induces somatic embryogenesis in heterologous tissues. So shown here are embryos on on leaves. So based on these data that baby boom has the ability to initiate embryogenesis on its own. It's not present in the egg cells, its expression, but it's expressed in the sperm cells. And after fertilization, it expresses only from the male genome. So this had led us to hypothesize, is it possible that this gene, under natural conditions, acts as an embryogenic trigger after fertilization? So in order to test that hypothesis, what we did was we expressed baby boom 1 in the egg cells where it doesn't normally express, and asked the question, would it induce parthenogenesis or embryo development without fertilization? So that's what exactly it does, is that when we express it in the egg cell, it results in embryo formation without fertilization. So shown here is a rice ovule, which was emasculated much before fertilization can happen, and you can see an embryo forming here. So baby boom 1 can induce parthenogenesis. It's egg cell expression. But to make a functional seed, you don't need only embryos, you also need the endosperm, which is the nutritive tissue. So we asked the next question, would it be possible that in these plants that we can have embryos that develop parthenogenetically, but the central cell still gets fertilized and forms the functional endosperm? So in order to test that, what we did was we took some of the seeds from these T0 transgenic plants, we let them form normally, undergo fertilization and all, and then we screened the T1 seeds from these plants. And what we can see is that 30 to 35% of our plants were haploids. It's because you're converting an egg cell directly into an embryo, but we can have still the fertilization of the central cell to make normal endosperm, so we can make normal seeds. Now, I'm not going to tell you why we need haploids. The previous speaker did a really good job at that. So we didn't leave it there, we took it further. So we looked at synthetic apomixis only, entirely from a sexual reproduction point of view. So in order to, be able to do apomixis or bring apomixis to a crop plant, you need to be able to bypass these two fundamental processes that are involved in sexual reproduction. One is meiosis, the other one is fertilization. Now, with the egg cell expression of baby boom 1, we can convert fertilization into parthenogenesis. But then, there is still the other process, which is meiosis, that needs to be somehow deleted or skipped. Fortunately for us, our collaborators in France, Raphaël Mercier, who was at INRA before and is at Max Planck now, and Emmanuel Guiderdoni, they showed it first, Raphael's group, first in the Arabidopsis, that a combination of 3 mutations, which is PAIR1 or SPO11, REC8, and OSD1, when we combine these are 3 genes involved in meiosis. You basically, if you mutate these genes, you can skip meiosis and convert meiosis into a mitotic-like division. So that's what stands for that's what MIME stands for. And you can produce basically diploid gametes, which have exactly the same genetic constitution that of any other somatic cell in the mother plant. So they're essentially clones of the mother plant. So then we ask the question is, if we combine these two different procedures together, which is MIME and parthenogenesis, can we produce clonal progenies? And we obviously used gene editing to knock out these MIME genes, and then combine that basically with our parthenogenesis strategy. So the expectation here is that the MIME will produce diploid gametes, unrecombined, unreduced, and then if they develop by parthenogenesis, we will end up having a 2N clone. But if they still undergo sexual reproduction and fertilization, then we'll end up having a 4N sexual progeny. That's precisely what happens, is that almost at the same frequency that we saw haploid induction in this line, when we transform it with this MIME gene editing construct, what we see is about 30% of the progenies were diploid and the rest of them were tetraploid. So diploids, in most of the grown or the cultivated rice, this feature on, which is very prominent in wheat, but it's suppressed in the cultivated rice, it reappears in tetrapolites. So that made basically screening very easy. So tetrapolites and diploids are phenotypically quite distinct, where we also obviously confirm them by flow cytometry to determine their genome ploidy. So, but how do you say the progenies that you got with this technology are actually the clones of the mother plant? So for that, what we did was we did the whole genome sequencing for one of the parent lines, and also progenies for the next 3 generations. Now, this is actually an inbred lab strain of rice, Kita-K. So there isn't much heterozygosity in this strain, but we were still able to find, SNPs that are scattered throughout the genome of this variety, and all of these SNPs were actually conserved across these generations, across these 4 generations. So no matter from which generation your plant comes, it has exactly the same genetic makeup as that of the original mother plant that it came from. But there are 2 bottlenecks at that point in this technology. One is the fixation of heterozygosity. That's what we're essentially doing, fixing that parental heterozygosity. Would that actually translate into fixation of heterosis or hybrid vigor? And at 30% or 35%, it won't be a technology acceptable in the field. So that had to increase to more than 80% to those agriculturally acceptable limits. So for this, we collaborated with Emmanuel Guidardoni at CIRAD, because we don't have rice hybrids and we don't have right to edit those rice hybrids, but CIRAD does. So this hybrid, which is a commercial rice hybrid, is mostly grown in this South America, in Brazil. It was developed by CIRAD. It is known for its high grain quality and low breakage rate. It's created by exactly the same cumbersome 3-parent cross. So we threw our Apomixis technology at this hybrid. So this is a really good example of what sexual reproduction can do and what fixing that with Apomixis can do. So shown here on the left is this is an FN hybrid, and it's allowed to undergo sexual reproduction, and you can see all these hybrid characteristics, they segregate. But if you fix it with this with synthetic Apomixis, there is no segregation. Everything looks the same. So indeed, the preservation of heterozygosity actually translates into fixation of hybrid vigor. You move it across any generation, it will still look the same. There is no segregation. The other thing that we also saw with this, we actually made some small adjustments in our constructs. We created a single construct, and with this single construct, what we also saw was that we can have apomictic progenies at about 99%. So that brings me to the end of my presentation. Just to explain how this technology works is that sexual reproduction involves 2 processes. One is meiosis, the other one is fertilization. Meiosis creates reduced and recombined gametes, and then fertilization basically brings a new combination of gametes, resulting in a recombined sexual progeny. But with synthetic apomixis, we use MyMay, which results in diploid gametes. There is no reduction, no recombination. And then with the expression of baby boom 1 in the egg cells, we can convert these diploid gametes into clonal progenies. Big thanks to all the people who have contributed to this work. And thank you for listening to me, and I'll be happy to answer any questions.
Thank you very much, Imtiyaz. I understand the talk now that I've seen it, a number of times, so it's good. I think I can actually give it now. But, it takes a few times, I think, to understand Apomixis, and really quite complex, I think, biology and genomics that went into this. So congratulations. I want to open the floor now to questions. We have, just under an hour, and I also want to but to congratulate everybody. I was looking at the number of people online, and there's still, I've got 3 screens of people the way I've got things out still, here online. I want to open questions, for open questions. I'm not seeing very many in the chat. I think that's because people are maybe writing them down and want to ask questions. So again, if I'll open the questions to, to any of the speakers or all of the speakers, just, put on your video and go ahead and ask a question and unmute your microphone. So and maybe raise your hand might be the way to get you. So are there any questions for the speakers?
Everybody was entirely too clear, apparently.
Something like that. I have many questions, so maybe I'll start. I can start to get people moving again. It seemed to work for Dave. This is a question for Jeff. I mean, if you look at the past, say, GMOs that were commercialized, it's been the big crops and a few traits. None of them have been really disease resistant. So why disease now, and what has changed?
So yeah, I think probably the reason for that is Dave touched on this previously, is that insects, herbicide tolerance, those are things that are encountered every year almost. I mean, if you have corn rootworm pressure, it's usually the same all the time. In contrast to that, if you look at disease, at least in corn in North America, I'll use as an example, is that you have differences in disease pressure. So it's not as consistent as you see with these other traits. And so typically it wasn't worth the deregulation journey with transgenics. But now, given our ability to identify dominant disease resistance genes and our ability to put them from a cisgenic standpoint from one crop from corn into corn, basically, it makes this journey a lot more attractive from a financial standpoint and, from a scientific standpoint. So I think that's a difference between now and previously, is that attitude has changed with our ability to do this stacking with disease, at least in corn.